Hello, sir. Good morning.
Dr. Ravi Shankar?
Yeah, Dr. Lakshmi, I'm ready.
Oh, you are here, right? We thought you are facing some network issues.
No, I was here. I said hi to Anuj also.
Okay. Then shall we start then?
Shall we start? Shall we get started then?
Yeah. We will, in a minute we will start.
Okay, 1 minute. I'll wait.
Okay. So let us get started. The committee, managing committee.
A very good morning to all. Welcome back to the second day of 7-day online workshop on method of data acquisition and analysis in NMR and HRMS, sponsored by DST SERB under Accelerate Vignan scheme. Now we are resuming to the technical session. We kindly invite your active participation. Our first honorable speaker is Dr. Ravi Shankar L. from Chemical Science and Technology Division, CSIR NIST. He is going to give us a lecture on the topic Structure Elucidation by 2D NMR Case Study. I humbly invite Dr. Janardhan Banoth, Assistant Professor, Department of Chemistry, NIT Calicut, to chair the session. Over to you, sir.
Thank you, Drisha. A very good morning to all of you. Today in this lovely morning session with us, Dr. Ravi Shankar Lankalapalli. He will be giving an overview on structure elucidation by 2D NMR case study. I formally welcome Dr. Ravi Shankar L to this workshop. It gives me immense pleasure to introduce our speaker, Dr. Ravi Shankar L. He completed his MPhil and PhD in chemistry from the Graduate Center, City University of New York in 2005 and 2009 respectively. From 2009 to 10, he worked as an adjunct assistant professor at the Queen's College, City University of New York. Later, he moved to CSIR, National Institute for Interdisciplinary Science and Technology, Trivandrum, as a DST postdoc and scientist. And at present, he is a principal scientist in the same institute. His research interests include isolation, structure elucidation, semi-synthesis and total synthesis of natural products, development of novel synthetic methods and pharmaceutically relevant molecules, as well as new chemical entities and synthetic carbohydrate chemistry. He is a very good researcher. He has many publications in highly reputed journals such as Organic Letters, Cellular Immunology, Organic and Biomolecular Chemistry, Journal of Carbohydrate Chemistry, etc. With these few lines about Dr. Ravi Shankar, I request Dr. Ravi Shankar to give a talk on the topic structure elucidation by 2D NMR case study. Over to Dr. Ravi Shankar. Dr. Ravi Shankar. Dr. Ravi Shankar. Can you hear my voice?
I think he got disconnected. Just we'll wait for 1 minute.
Yeah, he is not here.
He's not here.
He said he is facing some issues with his laptop. So
Okay.
he might need another 5 minutes if that happens.
Okay.
Dr. Ravi Shankar, can you hear my voice? Dr. Ravi Shankar?
Hello?
Yes, Dr. Ravi Shankar. Can you hear my voice now?
Yes, I can hear you. Sorry for the delay.
Yes, Dr. Ravi Shankar, I think I have introduced about you, and I think you can go ahead with the topic of structure elucidation by 2D NMR case study.
Thank you. Thank you. I would like to share the slides right now. Can I go ahead?
Yes, yes.
Kindly confirm if it is visible now.
Yes, sir, it is visible.
Okay. Thank you so much, Dr. Janardhan, Dr. Lakshmi, and Dr. Anuj, the organizers who gave me this opportunity to present during this workshop on NMR structure elucidation by 2D NMR in particular. So, as this is a workshop, can you please tell me, Dr. Janardhan, how many participants are there right now?
Now 110 participants are there.
Okay.
But around 200, the total number is around 200, sir, more than 200.
Okay, and most of them are research scholars, isn't it?
Yes, yes. Research scholars and MSc student as well as some of the faculty members are also there.
Okay, so I would like to get started.
Yes, yes, sir.
Sure. Thank you so much again, and good morning, everyone. Myself, Dr. Ravishankar Lankalapalli. I'm a principal scientist at CSR NIST. NMR structure elucidation is one of my key research interests as I work in the area of natural products, which involves isolation of natural products from both plants as well as microbial source. And NMR we use mostly during our synthesis as well. So today I would like to present some case studies from my lab on 2D. All of you have listened to the talks yesterday pertaining to NMR on physics aspects of NMR and also the NMR of peptides and proteins by a professor from University of Hyderabad, Dr. Boyan, I guess, and Dr. Kurur. And today it's going to be a little different. It's going to be on small molecules, and mostly I'll be presenting on the interpretation by 2D NMR. So before I present on that, I would like to stress on a very important aspect of what is the importance of NMR. Before I go into that, I would just give me 1 minute. I would like to open the pointer. Can you see my pointer?
Yes, yes, Dr. Ravi Shankar.
Okay, it will be helpful in explaining. So NMR spectroscopy, it has provided the foundations on which modern organic chemistry has been developed. And the advancements in organic chemistry depend upon the chemist's ability to interpret NMR spectra. This is a very important point, I feel, for the research scholars who are attending today. that the interpretation skills of solving the structure by NMR is very important, because it is not just to understand that you have, let's say if you are a synthetic organic chemist, and to confirm the structure if you have taken an NMR, it is not just a mere structure confirmation tool. There is a lot of many other factors which can give information about the molecules that we have synthesized or that we have been isolating. So, and this tool is an indispensable tool. Having experimental skills as a synthetic organic chemist, one must also have a very good strong interpretation skills, especially NMR. So that is the reason advancements in organic chemistry depend on chemists' ability to interpret NMR spectra. So we should inculcate the habit of interpreting the NMR and using it in our daily routine PhD life, and trying to get about the structure and its details and its three-dimensionality confirmation and its overall three-dimensionality of the molecule. You should try to be able to get it from the NMR spectra. So as this is a workshop and this is not like a seminar, so what I would like to do is I will talk in brief about some of the 2D NMR spectra that are routinely used in structure elucidation of organic molecules. So I will begin with the kind of 2D experiments that we commonly encounter during 2D NMR structure elucidation. There are two types. One is through bond. So through bond we have COSY, TOXY, HSQC, HMBC, and through space we have either NOSY or ROSY. Okay, so through bond, COSY, TOXY, HSQC, HMBC are very, very important experiments through which we will be able to do the structure elucidation. And once we solve the structure, if we want to know the three-dimensionality of the molecule and also the stereochemistry of the molecule or conformation of the molecule, we can take aid of NOSY or ROSY. Both are same experiments, just a slight variation. But both of them give the same result out altogether. So it is after solving the structure, you can get information of the three-dimensionality or conformation or stereochemistry of the molecule through space by NOSY or ROSY. So what I'm going to do is, before I go over the case study of how to interpret the 2D and how to derive the structural features or partial structures from the 2D, let us go into the understanding of what is meant by COSY, what kind of information does COSY give to us. COSY is a correlation spectroscopy. It is a very useful method for determining which signals arise from neighboring protons. And the correlations appear when there is a spin-spin coupling between protons. Okay, so we all are aware of the kind of parameters that we are supposed to use when it comes to a proton NMR. Okay, so in the proton NMR, other than the integration and the splitting pattern, we also get information about the coupling partners. That means the coupling protons within a chemical environment. Okay, so the ones which are within that same two different chemical environments, and these two chemical environments, when they couple with each other, there is a particular value called as a coupling constant, and that coupling constant gives information about the neighbor. So this is a very important information, and the same information we can get from correlation spectroscopy, also named as COSY, because it is not always possible to get coupling constant from a multiplet, because sometimes if it is not obeying the simple n+1 order rule, like if it is not a clean doublet or a triplet or a double doublet or a triple doublet, whatever it is, whatever the multiplicity it is, if we are not able to calculate coupling constant, maybe due to second order splitting or maybe due to overlap of signals, then we can take help of COSY experiments to get information on the neighbors, that is the neighboring chemical environments. Okay, so COSY is a very useful experiment which can give information about this signals which are spin-spin coupling on especially the protons. This is a proton-proton cozy that I'm talking about. And when the multiplets, especially when multiplets overlap, and when there is a second order splitting, then the 1D spectrum to get information about the neighboring environments is very complicated. So cozy is highly helpful in that regard. So this is a typical cozy spectrum where it is a two-dimensional spectrum, where we can see that there are cross peaks and there is a diagonal. So the diagonal is where the two protons in the x-axis and y-axis intersect. Okay, so for example, if you take let's say proton 4, that means proton on carbon 4. So if you take the proton on carbon 4, it intersects in the diagonal, and if you extrapolate it to the y-axis, you will find that around 4 ppm there is a same signal on the y-axis. So the same signal which is there on x-axis and y-axis, they intersect in the diagonal, and any peak which comes off diagonal is a cross peak. Any peak which comes off the diagonal is cross peak, and that is like a mirror image of on either sides of the diagonal. Okay, so for interpretation, we can take any one side of the diagonal and take those information of the cross peaks and try to derive information about the protons in the neighbor, that is either vicinal protons or even geminal protons. So here we are mostly interested in vicinal protons, that means protons that are separated by 3 bonds, or geminal protons which are protons separated by 2 bonds, that means 2 protons on the same carbon. Okay, so as this is a proton-proton correlation experiment, I repeat again that whenever you are unable to get a clear splitting pattern or a clear multiplet which is making you difficult to solve the coupling constant, then we can take help of cozy experiment to understand what is in the neighbor, which is in the vicinal proton, which is coupling with itself, or which is the geminal proton which is coupling with it. Okay, so that is the information about cozy. And the next experiment is HETCOR, heteronuclear. Just please hold on. I'm using WebEx for the first time, so I have some difficulty. Okay, the next experiment is called as heteronuclear correlation spectroscopy. So as the name implies, heteronuclear correlation spectroscopy, it is also a proton NMR spectra having a correlation, but this time it is having correlation with carbon NMR. So the carbon NMR is usually on the y-axis, which is also called as F1 axis, and the x-axis is called as F2 axis. So the y-axis is F1 axis and the x-axis is F2 axis. So the proton having a correlation with the carbon is nothing but information that we get on proton, which proton is contained on which carbon. Okay, so if you see a correlation, that means the correlation is nothing but a cross peak or a dot in this region, where you will find that by drawing a line on the x-axis towards the y-axis, and then drawing a line from that cross peak towards the x-axis, it means that this carbon is contained, this carbon is containing this proton. Okay, so this carbon is containing this proton, this carbon is containing this proton, and this carbon is containing this proton. So we can get information from HETCOR, that is heteronuclear correlation spectroscopy, about the protons that are contained on the carbon. So this is a direct single bond correlation. Okay, this is a direct single bond correlation, where there are other variants also, which I will explain in the next slide. But HETCOR has other experiments, so it requires that the compound quantity that is required in the NMR tube is slightly higher. So usually HETCOR is not something which is preferred. Rather, there is another experiment called as HSQC, which is a variant of HETCOR. So this HSQC, which is heteronuclear single quantum correlation, the name is not that important. What is the information that does it give? That is important. So this is similar to HETCAR, but this is more sensitive than HETCAR, where even with small amount of compounds, we will be able to get a decent 2D HSQC experiment. Okay, so but the information which it gives is again the same. That is, it is giving information about the protons that are directly connected to the heteroatom, that is carbon. Okay, so that is HSQC. So it gives information about single bond correlation. Now I'll talk about TOCSY. Now we have seen so far COSY, and which is a proton-proton correlation. We have seen HSQC, which is a proton-carbon correlation. Now let us look at TOCSY. TOCSY is nothing but it gives information which is similar to COSY, but sometimes it goes beyond COSY and gives information to us, and that is why it is called as total correlation spectroscopy. So the total correlation spectroscopy is about protons that are contained in one spin system. Let me explain what do I mean by spin system. Spin system means, let us take the example of sucrose. In this sucrose, you can see that there is a pyranose form, that is 6-membered ring, and there is a furanose form, that is 5-membered ring. In the pyranose form, the carbon number 1, 2, 3, 4, 5, 6, all these carbons have protons in continuation. Okay, so that constitutes one spin system. Whereas the 2' is a quaternary carbon, but 3', 4', 5', 6' in the furanose ring is having a continuity of carbons with protons in that carbons. So that constitutes the second spin system. So which means that this 6-membered is one spin system and the 5-membered is another spin system. And wherever there is lack of continuity, for example, 1 to 2', there is a lack of continuity. That means it contains an oxygen in between. Lack of continuity means that the spin system is switching over to the next spin system. So why this spin system is important? What is the role of it in the total correlation spectroscopy, namely TOCSY? In TOCSY, in COSY in particular, you will get information between carbon proton on carbon 1 and proton on carbon 2. Similarly, 2 and 3, 3 and 4, 4 and 5, and 5 and 6. But you will not get information of 1 with 3, because 1 and 3 are not vicinally located. Okay, if you look at the proton on carbon 3 and proton on carbon 1, they are separated by 4 bonds. They are not separated by 3 bonds. They are not vicinal protons. So in COSY, we have only a limitation that we can see, we can get information about vicinal protons only. But in TOCSY kind of an experiment, we can get information which is beyond vicinal, similar to this example which I just narrated. That means you get information about 1, 2, you get information about 1, 3, 1, 4, 1, 5, and 1, 6. But you will not get information about 1 and 2', because they are 2' is a separate spin system. Okay, so like that only you can see here, you can look at 3'. Okay, 3' corresponds to the furanose ring of this disaccharide. So in furanose, 3' is having a correlation with 6', 3' is having a correlation with 5', 3' is having a correlation with 4', 3' is having a correlation with 3', but 3' is not having a correlation with 1', because 3', between 3' and 1', there is a carbon called as carbon 2', which does not contain a proton. Okay, so 3' and 1', there is a carbon 2'. 2' does not contain a proton, so there is a lack of continuity. So that means that 1' is a separate spin system. Excuse me. So when there is a lack of continuity, that spin system breaks and we will not be getting signal. So that is why I have narrated that with the help of 3'. 3' can have a correlation with 4', 5', and 6'. Excuse me. That means 3', 4', 5', and 6' constitute one spin system. 1' is a separate spin system. 1, 2, 3, 4, 5, 6 is a third spin system. There are 3 spin systems in this molecule. So total correlation spectroscopy gives information about what are the spin systems and what are the protons within that spin system. That information we will be getting from total correlation spectroscopy. It is very important experiment. The next experiment is NOSY experiment. The NOSY experiment is also a proton correlation experiment. And as you are aware that NOSY is about the protons which are in the vicinity. that means the protons which are not connected through bond. So such protons which are in the vicinity are going to be showing up as signals in the NOC spectrum. And this is a 2D technique. NOC is a 2D technique. There is a 1D NOE also. We can do 1D NOE also by selectively irradiating the proton of interest. So here we can see in 2D, at a glance, we can see all the correlations that are available for the protons which are in the vicinity through the help of cross peaks for the protons that are associated in that vicinity. Okay, so these are not through bond, these are through space. I explained about HSQC already, which is giving information about the protons that are contained on the carbon. That means it is a single bond correlation. Next comes HMBC experiment. HMBC experiment is the most important experiment during structure elucidation. Okay, what is the information that HMBC gives? HMBC gives information about proton and carbon, which are separated by 2 bond and 3 bond. Any typical HMBC experiment gives information about proton and carbon correlation, which are separated by 2 bonds and 3 bonds. There are situations where proton and carbon, which are separated by 4 bonds, also show up as a cross peak in HMBC, but it is not that much reliable. But the most reliable information that you will be getting is protons that are separated by carbons by 2 bonds and 3 bonds. So let me explain that with the help of this terpene over here. So you can see here that there is a proton on carbon 1. So there is a proton on carbon 1. There is a proton on carbon 2. There is a proton on carbon 3. Let us explain this HMBC. Let us understand HMBC with the help of this proton on carbon 1. What do I mean by HMBC? 2 bond and 3 bond separation. It means that the proton on carbon 1 can have a correlation with carbon 2 and carbon 3. Okay, let me explain it once again. A proton on carbon 1 is separated from carbon 2 by 3 bonds. Remember, this is a proton-carbon correlation. A proton on carbon 1 is separated from carbon 2 by 2 bonds. Name again, a proton on carbon 1 is separated from carbon 3 by 3 bonds. Okay, so proton on carbon 1 is separated from carbon 2 by 2 bonds. A proton on carbon 1 is separated from 3 by 3 bonds. That means proton on carbon 1 can show a cross peak with carbon 2. Proton on carbon 1 can show a cross peak with carbon 3, but a proton on carbon 1 cannot show a cross peak with carbon 4, because that will be like 1, 2, 3, 4 bond separation. Okay, so a proton on carbon 1 can show a cross peak with carbon 2, can show a cross peak with carbon 3, can show a cross peak with carbon 8 because it is 3 bond separated, can show a cross peak with carbon 6, which is 2 bond separated, can show a cross peak with carbon 5, which is 3 bond separated. But a proton on carbon 1 cannot show a cross peak with carbon 7 because they are 4 bond separated. Okay, so like that, when you are getting additional information about the connectivity of the carbons and the protons and all that, this HMBC will give a huge amount of information as to how do we connect these small informations that we are getting from the prior experiments and the present data. Okay, so HMBC is one of the most important experiment during structure elucidation. Let me just take a 10-second break. Okay, I'm back, and as this is a workshop, I urge all the students and research scholars, whoever are attending, to stop me wherever you have a doubt, and whenever I'm not able to communicate properly or able to reach you properly. If I can see you, I know whether you're able to understand or not. So presentation, if you have any doubt. Are there any questions so far? Okay, shall I continue? Anybody?
Yes, sir. I think there is no questions in the chat box. I think you can continue.
Okay, so I'm coming to the main presentation of today, which is the case study of understanding the structural elucidation with the help of 2D NMR experiments that I have just narrated till now. Okay, so I'm taking few examples from my own lab where we have isolated a compound from the ethyl estrate extract of an actinomycete, which is actually a streptomyces species. Isolation of a natural product and structural elucidation of that natural product is one of the key research interests in my lab. And we do that with the help of 2D NMR. So I'm going straight into the NMR case study of isolation of a natural product, structural elucidation. So let us take a look at this proton NMR. And this is a case study. So in any case, when you have a structure ahead of you and you have to solve the structure, what you do is look at the proton NMR and try to classify what kind of a molecule it is. Well, as majority of you are, or I assume that you have the basic understanding of interpretation of a simple proton or a simple carbon, and I hope majority of you have the basic background in organic chemistry. So with that assumption, I'll move forward. If you look at this proton NMR, there is a not so complex NMR, but there are good number of peaks. So we have to solve this structure. Okay. So when you look at any proton NMR, the maximum information that you can derive from a proton NMR, you should keep it in hand and note it down. So what is that information I am getting here? With respect to the signals, I see that there are peaks from 6 to 8 ppm, which could be olefinic protons as well as aromatic protons. There are peaks between 3 to 4.5 ppm, which tells us that there are enough heteroatoms to which are bonded with carbons, and there is some methyl group because it is coming with 3 integration at 1.5 ppm. That means it is attached to an sp3 carbon, and there is another peak which is at 1.0 ppm, which is showing up as a doublet. So it means that that is also attached to an sp3 carbon. And there is a methoxy peak at 4.2 ppm, which means that this is a methyl attached directly to oxygen, right? So you can see that I'm getting maximum information from the proton NMR itself. Okay, and remember, structure solving is something which is a great skill. Whether you are an organic chemist or a biologist, if it is involving an isolation of a natural product, or if you are an organic chemist where you are intending to synthesize something but you synthesize it something else, and that something else, if you want to solve the structure with the help of 2D NMR, that can be a gateway to some major breakthrough. So having an understanding of structure elucidation is highly essential, a very good qualification, a skill which is indispensable, which will be helping you throughout your life during your research. Okay, so with that words, I'm continuing with this proton NMR, the kind of information that I got from the proton NMR, I have made some points. Okay, so after making the points, the next step in structure elucidation is to move forward and try to get information about the coupling constants. Okay, so coupling constants are very important piece of information, because when you measure the coupling constant, and what is essentially coupling constant? Coupling constant is giving information about which are the protons that are coupling with each other. Okay, that means there are two environments. Among the two environments, whether they are vicinal environment or geminal environment, if they are coupling with each other, it means that the coupling constant value is same for both. Okay, for example, if you look at this 5.47 ppm, it's a doublet. Okay, so we can calculate the coupling constant, which is the difference multiplied by the operational frequency, because in this case is a 500 megahertz NMR experiment, so multiplied with 500, you will be getting a coupling constant of 10 Hertz. Okay. Now there is a quartet here. In this quartet at 4.61 ppm, you can straight away calculate the coupling constant from the quartet, or it could be sometimes double doublet also. Okay, but at this moment I will just say it is a quartet because it appears like a quartet, and then try to calculate the coupling constant from that also. Okay, so then take that information and note down that information also on a piece of paper. So far I have showed you 2 slides only. The first slide was on the proton NMR. The second slide was on the coupling constants. Okay, I'm going a bit slow here so that you try to understand maximum of whatever I'm saying. So the first thing that I have done is try to see if you can do a broad classification based on proton NMR. I did the classification. I called, there is an aromatic group, there are heteroatoms, there is methyl groups. I did some classification, and then I counted the number of protons, identify the multiplicity and measure the coupling constants, and then try to identify the functional groups. Actually, through NMR and carbon, proton as well as carbon itself, you can identify many functional groups. There is no necessity to do IR. Okay, so with NMR itself, we can identify the functional groups also. Okay, now next is from the coupling constants, arrive at partial structures and identify protons which are exchangeable. So that means if there are some protons, by addition of a drop of deuterated water, and if that proton vanishes, then we can identify which are the protons that are exchangeable. Okay, here I have underlined one word, partial structure. 2 words, partial structures. Partial structures means what? Partial structures means trying to get maximum information about the structural features of the molecule. Partial structures. Once we get the partial structures, it is like a puzzle. Puzzle means there are different partial structures. With the help of 2D experiments, trying to club these partial structures so that overall it can give rise to the final structure. So again, I'm going back to the proton NMR. So this is the proton NMR. We have all the information that I have derived maximum from this molecule. So what is that? The broad classification has been done. We have a carbohydrate, we have aromatic protons, we have phenolic protons, because the phenolic proton is coming at somewhere between 9.5 to 10 ppm. So it's a phenolic proton. We have aromatic protons, and I'm at this moment I'm calling it as aromatic protons because they have heteroatoms. There are multiple heteroatoms to which the carbons are attached. They can be oxygen, or they can be nitrogen or sulfur also. So I will just consider I'll just go with an assumption that there may be a carbohydrate in this because of the characteristic peaks which are coming in the region of 3 to 5 ppm. So there are 4 exchangeable protons which have exchanged with D2O, 3 from the carbohydrate region and 1 from the phenolic region at 9.78 ppm, that also exchanged by addition of D2O. So there are 4 exchangeable protons. There are 2 methoxy groups in the carbohydrate region. So one is coming at 4.2 and the other one is coming at 4.22. And then there are 2 methyl groups. One is CH3 attached to a quaternary carbon. Quaternary carbon, why it is quaternary carbon? Because CH3, if it was attached to a CH proton, it will appear as a doublet. But it is appearing as a singlet, so we can conveniently say that it is attached to a quaternary carbon, and that too an sp3 carbon, because if it was an sp2 carbon, it will be allylic, and that will come in the region of 2 to 2.5 ppm. Okay, so this is an sp3 carbon only, and that is CH3. And then there is a CH3 attached to CH, because this CH3 is appearing as a doublet in the region of 1.2 ppm. That is why this can be considered as CH3 attached to CH only, which is also an sp3 carbon because of the chemical shift region. Okay, so the chemical shift region also is very important to understand that the chemical shift, you should be able to predict the NMR of any molecule given the structure, and predict the NMR based on the chemical shifts, right? So with the help of the chemical shift information, we can tell very conveniently that the CH carbon is sp3. And there is also a presence of a terminal alkane, because terminal alkane functionality clearly shows up in the NMR with a 3 proton integration altogether, where 5.46 ppm at having 1 proton is having a coupling constant of 11, 6.15 having a proton with a doublet of J is equal to 18, and then there is a 6.95, one more proton which is appearing as a double doublet having a coupling constant of 11 as well as 18. Okay, that means 11 means 11 hertz is having coupling of 6.95 with 5.46, and 18 is a coupling constant where 6.95 is adjacent to 6.15. Okay, so with the help of coupling constants, we can easily derive that there is a presence of terminal alkene. Next, we will move on to the carbon NMR. Now, the same information, whatever you have derived from the proton, we should try to arrive from carbon as well. So from the carbon also, we have some carbohydrate region, which usually comes in the region of 70 to 80 ppm. We have a bunch of aromatic carbons, which comes in this region. And there are some carbons which is in the region of 150 to 160. So there may be presence of carbonyl groups in this region. Okay. So again, try to see if you can do broad classification based on carbon NMR. Count and assign the labels to carbons. Now it is the time to give some labels to carbons. You can call it as 1, 2, 3, 4, 5, 6, or you can call it as A, B, C, D, E, F, G, H. Whatever it is, you just label the carbons, either starting from the left side to the right side or from right side to left side. I'll go back to the carbon NMR. Start labeling the carbons from right side to left side, or start labeling the carbons from left side to right side. Okay, what do I mean by labeling? Just give an annotate each carbon, right? Like 161.27 is, let's say, I'm labeling it as carbon 1. 158.70 is carbon 2. 154.89 is carbon 3, so on. Okay, so that is labeling, annotation. After assigning the labels to carbons, why we are assigning the labels to carbons? Because we have to finally find out which are the carbons which are interconnected. Okay, so that is why we have to label the carbons and then identify the functional groups, which we have done already. We said that there is a carbohydrate, there is an aromatic system, there is a carbonyl system, and all that. And try to arrive again, if possible, partial structures also from carbon NMR, just like the way you have done from proton NMR. So presence of carbonyl, presence of peaks in the region of 70 ppm indicating the carbohydrate region, and presence of polyaromatic system because of bunch of aromatic carbons. Now coming to depth 135 experiment, this is actually not a 2D experiment, it's a 1D experiment. Depth 135 experiment gives information about carbon signals which are methyl, methine, and methylene. Okay, methyl means CH3, methylene means CH2, methine means CH. Usually in depth 135 experiment, the methylene carbons show up in the negative phase, that is the bottom region. The methyl and methine, that is CH3 and CH, appear in the positive region of the 1D NMR experiment called as depth 135. So here on the top you will get information about CH3, CH carbons. Bottom you will get information about CH2 carbon, but it doesn't give any peaks corresponding to quaternary carbons. So we can easily identify with the help of depth 135 from the carbon NMR that how many quaternary carbons are there. So identify the quaternary carbons with the aid of depth 135 as well as 13 carbon C NMR, and identify the methylene carbons which are present below, and identify the methyl and methine carbons which are present above. So by doing so, by looking at carbon NMR and depth 135, we found that there is a presence of 13 quaternary carbons. Okay, one methylene carbon is there, that confirms the presence of terminal alkene moiety. Okay, now you see this point number 10. The points that I made here is all about the information that we have received so far from interpretation from proton and carbon itself. Okay, if you look at 10th point, I was mentioning earlier that there is a terminal alkene, right? So the terminal alkene is nothing but there is a CH2. Okay, though both the protons are in two different environments of the CH2, but the carbon is the same. And that carbon is attached to, is actually an sp2 carbon. So an sp2 carbon usually comes in the region of 110 to 130 ppm. Okay, all these aromatic carbons, polyphenic carbons, they come in the region of 110 to 130 ppm. So indeed, this 117.03 is a carbon which is a methylene carbon, and it is having two protons that are geminally associated. Okay, so this is a confirmation that we do have a terminal alkene moiety. Now let us see what are the points that I have gathered so far. I have taken a broad classification. I have done D2 experiment to identify the exchangeable protons. From the proton NMR, we identified that there are two methoxy group in the carbohydrate region, and there is one quaternary carbon attached to sp3 and another CH3 which is attached to CH. There is a terminal alkene moiety. There is a presence of carbonyl. Peaks in the region of 70 ppm indicates a carbohydrate ring. Peaks in the region of 110 to 130 ppm indicates a polyaromatic nature. And then there are quaternary carbons, 13 number, and one methylene carbon, which confirms the terminal alkene moiety. So you can see that we gather maximum information from the partial structures or from the proton NMR and carbon NMR itself. Okay. So what are the partial structures that are derived so far? Pictorial representation: carbohydrate, terminal alkene, phenolic system, methoxy, methyl attached to sp3 carbons. One is quaternary, one is methyl, and then carbonyl. So this is the pictorial representation of the partial structures that are derived so far from proton and carbon itself. Now comes HSQC. Okay. HSQC experiment, I told you earlier that HSQC experiment is information about the protons that are contained directly on the carbon. That is, HSQC is a single bond correlation. Okay, so you remember that I was asking you to label all the carbons A, B, C, D, E, F, G, so on, or 1, 2, 3, 4, so on. Whatever labels you have done on carbons, this is a time where you can transfer that labels to the protons to which those carbons are attached. Okay, for example, if you look at this, cross peak, right? If you look at this cross peak, this is a carbon. Let's say this is carbon 22. Okay, during my labeling, I identified that this is carbon 22. So just draw a line straight to the cross peak, right? And then extend that line towards the x-axis. You will find that this carbon is associated with this proton, or this proton is associated with this carbon. This proton on the x-axis is associated with this carbon. So HSQC gives us an opportunity to transfer the labels of the carbons to the protons, right? So if this carbon is labeled as let's say 18, then this proton signal is 18. If this carbon is labeled as 22, then this proton signal is 22. This is the acetone solvent peak, because acetone is the solvent that was used. It's a residual solvent which comes in the NMR. Okay, these are all important, where we are getting information about protons that are contained directly on the carbon through single bonds, which gives us an opportunity to transfer the labels from carbon to the protons. So identify the protons associated with the carbons and transfer labels from carbons to protons as HSQC gives information of single bond correlations. That is what I have just said. Protons which are not assigned to any carbon can be identified as the protons which are associated with heteroatoms such as oxygen or nitrogen. Okay, the partial structures derived from proton can be verified from carbon NMR after label transfer from HSQC, and now generate a table of data so far that has been arrived. This is the table that I am talking about. That means if there is a carbon which you have labeled as 24, which is labeled as 2, which is having a chemical shift of 24.2, then That 24.2 is a carbon which is containing a proton which appears in the proton NMR at 1.34 ppm. Okay, I'll repeat again. If there is a peak in the carbon NMR as 56.9, which is labeled by you as 3, then that 56.9 labeled carbon is having a proton which is coming at 4.26 ppm in the chemical shift of for the chemical shift of proton NMR. Okay, and the integration tells me that 4.26 ppm is having a 3 integration. That means there are 3 protons associated with that signal, and it's a singlet. And if it was having some other multiplicity, then you can calculate the coupling constant also and generate a table like this. Whenever you look at any journal of natural products or natural product related publications, you will find that there is a table which contains information about each proton, each carbon, its splitting pattern, everything will be there. Okay, so generating a table is very essential when it comes to solving the structure. And with this table only, we will be moving forward to the next level of structure elucidation. Now that I have labeled all the protons which are there in the proton NMR, so we have to now take help of COSY experiment. Remember that COSY experiment gives information about protons that are vicinal protons or protons that are geminal protons. 2 protons in 2 different environments. The relation between these protons within in the 2 environments, namely vicinal protons or geminal protons, that information we will be getting from COSY. Okay, try to find out from COSY maximum information. That means, for example, if this is a CH proton, assume this is a CH proton. Okay, and you see that CH proton, which is assumed that it is labeled as 6. Okay, that 6 is having a COSY correlation with this doublet, and this is let's say labeled as 5. So 6 and 5 are vicinal to each other or also geminal to each other, but if it is geminal, we can find out easily from the coupling constant, because geminal protons on an sp3 carbon have a very large coupling constant of 12 to 16 Hz. Therefore, with the help of coupling constant, we can find out whether they are geminal protons or vicinal protons. But I'm telling you, this is vicinal proton. That means this proton is associated with this proton. That is why you can see that there is a correlation here in the form of a dot of the diagonal, and which is called as a crossbeam. Okay. Identify the protons which exhibit either vicinal or geminal coupling, which I was just mentioning, and then arrive at partial structures from the COSY. Arrive at maximum partial structures from the COSY. So this is about the partial structures. Sorry, I lost my pointer. Let me open it again. Can you see the pointer?
Yes, sir.
Okay.
I think again the pointer has lost.
Okay. Can you see the cursor?
Yes.
Cursor, I'm moving the cursor, no?
Yes, sir. Yes.
Okay. Okay. See, look at these partial structures that I have arrived. These are the partial structures that I got from COSY. So there is a partial structure of 1 to 5, and there is a partial structure of 5 to 9. There is a partial structure of 9 with X2 and all that. So you can see that there are many partial structures that I am having over here, which I derived basically from COSY. Okay. So what does it mean? One is vicinal, one is having a coupling with 5. 5 is having a coupling with 9. 9 is having a coupling with X2. X2 means I labeled it as X2 because it is associated with this. It is an exchangeable proton which is associated with a heteroatom. As it is not a carbon, this is labeled as X2. Okay, so X can be oxygen, nitrogen, or sulfur. So this is the partial structures that we got from the COSY experiment. Similarly, we got another information. We got 8 with 7 and 7 with X1. Now, can we derive a partial structure with this COSY information itself? You can see that 1 is next to 5, but 5 is next to 9. So that means 1, 5, and 9 are in series. And 9 is next to X, which is attached to X2. So that means 9 is attached to X, which is X2. You can see that this partial structure information we got from the COSY correlation data that we got from here. Similarly, 8 is next to 7. 7 is next to heteroatom, which is attached to X1, which means that 8, 7, and 7 attached to X, X attached to H. So these are two partial structures that we got from COSY experiment. And after getting those partial structures we actually got from COSY correlations. That means we combined the COSY correlations to get little bit of more information about the partial structures. Okay, so here we have a proof of terminal alkene. I will show it to you in a bit. Here we can see that there are aromatic protons which are having 8 Hertz coupling constant. Usually aromatic protons, when they are having 8 Hertz coupling constant, that means that they are orthocoupling. Similarly, when there are two aromatic protons which are associated with each other with the 1 Hertz coupling constant, then they cannot be separated by 2 bond. Rather, they are separated by 4 bond. So you can see that sometimes we see allylic protons, meta coupling protons have a very small coupling constant of 0.5 to 1.2 Hertz. So that is the case here, where 13 and 16 are thought to be vicinal protons, but they are not vicinal protons. They are having a long range correlation separated by 4 bonds that is happening, that is derived from the help of the coupling constant that we have here. That means there is a meta coupling between 13 and 16. There is an ortho coupling between 11 and 21. There was also a correlation of a partial structure between 15 and 22. That is very clear because we already know that we have a terminal alkene moiety from our proton NMR itself. So let us look at the partial structures that have been derived so far from the COSY experiment. This is one partial structure. This is a second partial structure. We have an ortho coupling, we have a meta coupling, we have a terminal alkene. Okay. Now comes the most important experiment, the mother of all, the 2D NMR experiments, which is called as HMBC. HMBC gives information about proton and carbon which are separated by 2 bonds and 3 bonds. Okay, I repeat again, HMBC gives information about protons and carbons that are separated by 2 bonds and 3 bonds. Okay, so as we have already labeled the carbons and we transferred the labels to the protons, we can take a proton, any one proton. For example, if this proton is taken, okay, this proton, let's say it is labeled as 6. Okay, if this proton is labeled as 6, that means it is a contained on carbon 6. That's what it means. This proton is contained on carbon 6. So that proton contained on carbon 6 is having a HMBC correlation with let's say 5, and this is also having HMBC correlation, let's say, with 23. Okay, that means this proton is having, which is contained on carbon 6, is having a correlation with carbon 5, is having a correlation with carbon 22. That's what it means. I will show another example. Let's say this is a proton which is labeled as 27. The 27 is a proton which is present on carbon 27. So proton on carbon 27 is having a HMBC correlation with, let's say, 18, and this is let's say 15. Okay, so this proton having a HMBC correlation with this carbon, it can be HMBC correlation with this carbon. But we don't know whether it's a 2-bond correlation or 3-bond correlation. Okay, so that carbon to which this proton is associated can be 2-bond or can be 3-bond also. Okay, sometimes both can be 2-bond or sometimes both can be 3-bond also. Okay, that information we don't know. We will arrive at that information also shortly. Now connect the partial structures using HMBC correlation data. Okay, so the data has been received so far from proton, carbon, HSQC, all that information, whatever data we have received so far, we tabulated it. And we are now further expanding the tabulation by introducing HMBC information also, where in the table of data we have informed in the table we are getting information from the table that label 5 means carbon 5. Carbon 5 proton is having HMBC correlation with carbon 1, or proton on carbon 6 is having HMBC correlation with carbon 2. Proton on carbon 14 is having HMBC correlation with carbon 10 and carbon 11. Okay, that's what it means. So now combining partial structures with the aid of 2D NMR, which is called as HMBC. So here I have given you, for the sake of simplicity, I have given you small information here. This A, D, B, C, basically it means that I want you to follow me, as we are not meeting face to face. So that's why I have given some labels here. I want you to follow A first, and then B, and then C, and then finally D. Okay, let's not go to this table. Let us look at partial structures that we have so far. So this was one partial structure, 159X2. This is a partial structure which we got from COSI. This is another partial structure which we got from COSI. This is a terminal alkane partial structure which we got from proton NMR itself. And this is another information of methyl contained on a quaternary carbon attached to an sp3 carbon. That is why it is a singlet, and it is appearing in the region of 1.5 ppm, 1.2 ppm something. So these four information we have: this information, this information, this information, and this information. Now with the help of HMBC, we should try to connect. Can we see there is a relation between all these partial structures or not? That is what we should see. Let us see. Now A means this is a table which I am having for HMBC. A is proton on carbon 5 is having HMBC correlation with carbon 1. Well, we can see that 1 and 5 are separated by 2 bonds, so that means it is correct. So this is one correlation. Next is proton on carbon 7 is having HMBC correlation with 9. Okay, proton on carbon 7 is having HMBC correlation with 9. But 7 is a part of this partial structure. 9 is a part of this partial structure. They are not connected. Okay, unlike 1, 5, 7 is here, 9 is here. Now this HMBC is giving us an opportunity to connect these two partial structures. Okay, now what did I say? It is a proton on carbon 7 connected to carbon on carbon 9, which is connected through HMBC. Now remember that this proton on carbon 7 is a double doublet. Why it is a double doublet? Because this proton is coupling due to this proton 8. This proton is also coupling due to this proton x1. That means it's a clean double doublet. If there is a carbon, assume if this carbon is directly attached to 9, which is a 2-bond correlation between this hydrogen and that carbon, then there is already a proton there, which means it cannot show double doublet. Similarly, if you take a look at carbon 9, it's having a proton. Okay, that proton is showing up as a doublet due to this proton, or sometimes due to that proton. We don't know, but it is a doublet. Okay, if it was having a coupling with this carbon also, then due to the vicinal nature of 9 with 7, this proton and this proton also should couple, but they are not, which means that there may be another carbon in between. Okay, so that carbon could be carbon 6. Why it is carbon 6, I'll explain later. Now you can see that B is a proton having a proton on carbon 7 is having HMBC correlation with carbon 9. Okay, so proton on carbon 7 is having HMBC correlation with carbon 9, and they are 3 bonds separated. If it was 2 bond, then it should be C7 and C9 directly connected, which is 2 bond correlation, and that is not possible because of the coupling constant, because of the splitting pattern, because of the multiplicity. Okay, so that means they must be connected through 3 bond, and there may be some carbon in between. If it was a heteroatom, On the other hand, if it was a heteroatom, it is already having a heteroatom, and if there is another heteroatom, this carbon is this proton on carbon 9 will be highly downfilled. So it is unlikely that there is a heteroatom. There is only carbon in between, and that is a carbon between carbon 9 and carbon 7. Okay, now let us look at C. 9 is having a correlation. That means proton on carbon 9 is having a correlation with 1, X3. What is 1? 1 is already there in the partial structure, so that information is not helpful for us, but it is having a correlation with X3. X3 means it cannot be this one because it is already X2, and it cannot be this one because it's already 5. So there must be another heteroatom on this carbon, which is X3. That is the only way we will be able to justify the proton on carbon 9 having a correlation with X3. Okay, that means carbon is associated with a heteroatom which is connected to X3, an exchangeable proton. That means this proton and this proton X3 and this carbon 9 are 3 bonds separated. They are 3 bonds separated. So this partial structure is correct. Okay, and remember I was mentioning that this is a carbon. This is a quaternary carbon. It is if it was not a quaternary carbon, that means if it is either a methylene or a methine, okay, then we will not be having a heteroatom here. Okay, if it is a methylene, it cannot be having a heteroatom. If it is a methyl, it is impossible. Then there is only 2 options. Either it can be CH or it can be quaternary carbon. It cannot be CH because if there is CH, there should be splitting. So this is quaternary carbon only. So that sp3 carbon, there is only one option left to satisfy the 4th valency. That is nothing but it should be having 2. Okay, it should be having 2, which is methyl carbon which is having 2 as the label, methyl carbon. And already we have seen that methyl carbon is the carbon which is associated with a quaternary carbon, which is sp3 carbon. Okay, so 6 is sp3 carbon and 2 is the methyl. So that is why you can see that there is a 2 bond HMDC correlation between 6 and 2. Okay, so this is a very important partial structure that we have got. Can we get more information from this? Let us see. So this is the information that we have so far, partial structures, and we told already that there could be a carbohydrate. Therefore, is there a possibility? Now starts assumptions. Okay, during structure elucidation, there is maximum chance that you will be having you will be forced to do assumptions. You have to do assumptions, and that assumptions should be logical. You cannot make wrong assumptions. Your assumptions should be logical, so that you will narrow down your assumptions to possibilities of correctness. Okay, so to get the possibility of correctness, your logic should be very important, so that your assumptions are correct eventually. Okay, so let us assume that this carbon 5 and carbon 8 are having ether bond, so that it can satisfy my requirement of presence of a carbohydrate moiety. So with that assumption, I will move forward, and my assumption is logical, because I can see that if C5 is attached to CH3, which is 1, it makes sense, because I already told you in the past that there is a methyl group in the aliphatic region. In that aliphatic region, that methyl group is associated with a CH, which is methine. That is why it is appearing as a doublet. So this methyl, which is labeled as carbon 1 here, is associated with carbon, which is actually a methine. That is why the CH3 is appearing as a doublet. Okay, now if this carbon is associated with some other carbon, okay, it is impossible, because it is having a heteroatom. Okay, and then this carbon is also associated with that heteroatom. To complete the cycle, I have given this as a 6-membered carbohydrate moiety. Now we will move on to the next partial structure, which is a terminal alkene. So what is the information that I have about terminal alkene? There is a 15 and 15 prime. There is a carbon which is 22, but 15 and 15 prime is not associated with 22. It is associated with 23 in HMBC. Okay, so let us see what is 23. 23 is actually an aromatic carbon. Yeah. Before going into the terminal alkene, let me dwell a little bit more on carbon 8. Okay. Now carbon 8, because I am telling that it is having a carbohydrate, there are two possibilities. One is it can be a O-glycosidic linkage. That means carbon 8 can be associated with oxygen as a glycosidic linkage, or it can be a carbon linkage. It can be a C glycoside. Okay, so carbohydrate means there is O-glycoside or C glycoside, but chances of being a N glycoside or having a sulfur glycoside are quite rare. So we will just go with some assumption over here again. It could be O glycoside or C glycoside. If it is O glycoside, there must be an oxygen here on the carbon 8. So if there is an additional oxygen on carbon 8, it should be appearing in the carbon NMR at above 100 ppm, around 100 ppm. But what is the carbon 8 chemical shift in carbon NMR? It is only 76.2. So it tells us that we have a CC carbon linkage. That means it is a C glycoside, but not an O glycoside. Okay, so that information with the help of simple chemical shift itself we are able to get. Okay. So O glycoside is wrong. C glycoside is correct. Now let us move on to the next correlation. I have given here that there is a correlation of 8 with 20. Okay, you can see 8 with 20 there is a correlation. 8 is what? 8 is already a part of the partial structure which we have earlier from our carbohydrate. 8 is a part of the partial structure which we have from our carbohydrate, and 8 is having a correlation with a quaternary carbon labeled as 20. But 20 is actually an aromatic carbon, which is because it is having a chemical shift of 129.8, which means that there can be an aromatic moiety. The first aromatic ring can be here. It can be heteroaromatic or it can be simple carbon aromatic only. So 8 can have a correlation with 20. 8 can also have a correlation with this 20 because this is a 3-bond correlation and this is a 2-bond correlation. So either this is the correct structure or this is the correct structure. I don't know, but I'm just going with the assumption with these two partial structures. And coming to the other terminal alkene, and the terminal alkene HMBC information I have here, where I have information that 15 prime is having correlation with 23. 23 is also another aromatic quaternary carbon. 15 prime can only have correlation with 23, means that because this is the longest correlation of 3-bond correlation with this proton with this carbon 23. Okay, so this proton can have quaternary carbon 23 HMBC correlation. So this is the only possibility that we have. Now as the time is running, I will just move on with all the partial structures information that I have, and I will arrive at all the partial structures that we have in a nutshell. Okay, so we have this aromatic moiety, this aromatic moiety, we have this aromatic moiety which is associated with methoxy or this one, and this phenolic moiety, and this carbohydrate I showed you before already, which is possibility of having this structure or that structure. So at this moment, you can stop doing your structural elucidation to find out whether this is already a known natural product or an unknown natural product. So take an advanced structure, partial structure from the list of partial structures that you have, and then go to a database such as SciFinder, draw the structure, put it in the SciFinder. With a substructure search, you can do the searching, and then you find that there are 18 hits over here by doing that partial structure analysis. And then we find that these are the possibilities that are available out there, and with the help of other partial structures, we narrow down to this molecule. This is the possible structure and this is the right structure, and it turns out that final elucidated structure is a known natural product, and this is the molecule of the final known natural product. So with this, I will ending my presentation because I don't have time for talking about the second case study, which is on synthetic organic chemistry. And as my time is up, I will be now happy to take questions from you. Thank you.
Now the presentation is open for the discussion. If you have participants, if you have the questions, you can write in the chat box, or you can directly ask to the speaker.
I don't have the chat box open, so I request you to kindly read out the question to me, please.
Yes, yes. So, sir, there is one question from Liaquat Ali. TOSI case, you talk about a different spin state. Will you please enlighten about spin state, how it is different?
Okay, I'll go to the TOSI. This is the slide I was talking about. So let us look at this slide. Okay, so this is a sucrose molecule. It's a disaccharide. Here you can see that there is a 6-membered pyranose and there is a 5-membered furanose. Let us take a look at 6-membered pyranose. TOSI is giving us information about all the carbons having protons in continuity, which means that if you look at carbon 1, it is having a proton, 2 is having a proton, 3 is having a proton, 4 is having a proton, 5 is having a proton, 6 is having a proton. Okay, so which means that all these protons are in continuity, they are in continuity. But if you look at 1 and 2 prime, the continuity is lost. That means carbon having proton, carbon 1 having proton, carbon 2 prime is a quaternary carbon. So we lost the continuity. So wherever there is a continuity in vicinal nature, continuity of vicinal nature as well as geminal nature, only vicinal nature continuity constitutes one spin system. That means whatever I labeled here, 1, 2, 3, 4, 5, 6 is one spin system. 3 prime, 4 prime, 5 prime, 6 prime is another spin system. 2 prime does not have any proton, so it is not a spin system. 1 prime given here as CH2 is a third spin system. Okay, clear?
Hope this question is clear. Sir, there is one more question. There are few questions. I'm going to read it out. So from Nikhileshwar Reddy, sir, how can we quantify using NMR?
Quantification of NMR can be done with the reference compound. For example, if you are having a, let's say you are doing a synthetic experiment and you obtained the pure product. The pure product basically is actually quantified from a mixture. Okay, so one of the major main requirement of quantification through NMR requires you that you should have a pure compound. So once you have a pure compound and you know you have the pure compound with you, then with the known concentration you add some pure compound along with a reference compound. Okay, the reference compound can be something like having a characteristic methyl signal. Okay, so when you have a reference compound which is a known amount, a pure compound also with a known amount, then altogether if you are taking an NMR, it will give a splitting, it will give a NMR. Okay, now add the same amount of reference compound in a mixture, right? Then you do the integration, and with the help of integration we will be able to find out whether what is the quantity of the of the compound present in the mixture. So for quantification you need to have pure compound and a reference compound. First you have to run a proton experiment with the pure compound and the reference compound with known concentration, and do the test experiment where the reference compound in known amount is added into a mixture, and then with the help of integration you should be able to figure out.
Okay, sir, I think this may be clear. And the next question is from Sivagni. Is it possible to obtain positive and negative peaks simultaneously in NOSI, proton-proton NOSI? And if so, does it help in structural elucidation?
No, answer is no. If you want to get structure elucidation information in NOSI, first of all, NOSI is an experiment where you should be taking help of NOSI only at the end after solving the structure to get information about the three-dimensionality of the molecule or the vicinity of certain groups in the moieties in the molecule. Okay, so if you are planning to get information about NOSI through 2D, first of all, one requirement is that you can have the structure solved with the help of COSY, proton, carbon, HSQC as well as HMBC or TOXI also in particular. Then only you try to use the NOSI experiment data to solve the structure or get information about the three-dimensionality. There is another experiment which I can suggest, which is called as 1D NOE. 1D NOE means you will not be getting a diagonal like that. You will be getting, you will be irradiating a proton of interest only one proton of interest. Because in 2D NOE, you will be getting information about all the protons in the vicinity of the whole molecule. But if in 1D NOE, we can selectively irradiate one proton of interest and then find out it is having a NOE correlation with which proton in the vicinity. Because in 1D NOE, the proton which is irradiated will come in one phase and the protons in the vicinity will come in a opposite phase. Okay, so 1D NOE is a very useful experiment in structure elucidation.
Okay, so the next question is from Anbajagan. How to determine X atom other than hydrogen and carbon atom?
Very good question. Actually, let us assume here we have oxygen. Okay, you try to derive the maximum proton count from the proton NMR and the carbon count from the carbon NMR, and take a high resolution mass spec where you can get some, where you can come to know about the exact mass of the molecule. Then by doing some math, we can find out what is the heteroatom that is present. But assume you have nitrogen, because oxygen is oxygen 16 and it is not NMR active nucleus, right? So nitrogen also, we have nitrogen 14, nitrogen 15. So there is one experiment called as proton nitrogen HMBC. Okay, so if you can write down, it's a proton nitrogen HMBC. So far we have seen proton carbon HMBC. There is a proton nitrogen, proton N15 HMBC. So if you have nitrogen in your molecule, you can have correlation of proton with nitrogen. Because if you want to take directly nitrogen experiment, an N15 experiment, you require a large amount of compound and you will not be getting any information at all. Rather, a sensitive technique is called as proton N15 HMBC, which gives information about the heteroatoms such as nitrogen in the molecule. So proton, carbon, and NMR experiments alone is not sufficient to find out what is the heteroatom that you have. You must also have HRMS, which gives information about the exact mass of the molecule.
Yeah, I think there is no more question in the chat box. Is
Okay.
control room, is anybody raised hands? I think if there is no more question, thank you very much, Dr. Ravi Shankar, for your wonderful talk on structure elucidation by 2D NMR. Hope all the participants have understand all the different type of experiments and how to utilize to identify the structures. Over to control room.
Thank you very much, sir, for the very detailed explanation on 2D NMR. Thank you. The second session will start within 5 minutes. Please be online.
Thank you again. Thank you so much.
Hello, I hope I'm audible. This is Bharatwaj.
Yes,
Yes, sir,
Dr.
you are
Bharadwaj,
audible.
Yeah. I'm just trying to see whether my this thing will work. My tablet will work. So should we test it now?
Yes, yes. You can go ahead.
Because I don't seem to have share presentation or anything
Yeah,
like
yeah.
that.
You have two equipments connected.
Right devices.
Yeah, yeah.
Right. I wish I can use my tablet because I might end up writing a little bit. Let me see whether I get the opportunity to do it.
Yeah, is it? You got the permission?
In the computer, yes, but in tablet, if you could give, I would appreciate that a lot.
Okay.
So you would see a second login for me.
Yeah.
That has a tablet. This thing, I would appreciate if you could give me there.
Yeah, it's not giving me permission.
I got it.
You
I got it. I'll try now. Let me see how this works. Okay. Share screen. Start broadcast.
Yeah, it's coming.
Yeah. So
Yes,
I
yes.
hope the slides are visible
Yeah,
now.
it's visible.
Yeah, unfortunately, it's not updating fast enough.
Okay.
Is it coming up? Because I'm able to see my own screen where I'm trying to show the screen coming up, but unfortunately, that's not showing up at all.
Yeah, it's visible. Actually, it's changing also. For us,
It's
it
changing?
is fine. Yeah,
Interesting. So you're able to see the white screen and then the spectrum and protein coming up?
Yeah, protein structure is there. Yes.
Okay, very good. So this is why I have a separate screen open on my computer. So the video will come from my laptop while I'll be writing here. This makes my life a little easier. Okay, perfect. I think I'm all ready. I'll wait until I am asked to talk.
Now we can move on to the next session by Dr. Bharatwaj Satyamurthy from Department of Chemistry, ISO Bhopal. He is going to deliver a talk on the topic Application of NMR Chemical Shift Towards Characterizing DNA-G Quadruplexes. It is my pleasure to welcome Dr. M. Shankaralingam, Assistant Professor, Department of Chemistry, NIT Calicut, to chair the session. Welcome, sir.
Thank you, Tricia, for your words. So, a very warm good morning to all of you and welcoming you for this today's second session. So now let me formally welcome the speaker, Dr. Bharatwaj Satyamurthy. It gives me an immense pleasure to introduce Dr. Bharatwaj. He has obtained his MSc from IIT Madras and PhD from State University of New York, Buffalo. And right after his University of Michigan and Duke University for his postdoctoral studies. Then right after his postdoc, he joined as an assistant professor at ISER Bhopal. And presently he is associated there, and his research interests include NMR spectroscopy, biomolecular structure, nucleic acids, and G quadruplexes, and conformational dynamics, and NMR methods and development. So based on his research accomplishments, he has published several interesting articles. So to name out few, he has published two Nature and 3 Nature Structural and Molecular Biology and JAX. So over to you, sir.
Thank you very much. I hope I'm audible.
Yes, you are audible. Yes.
All right. Good. Let me get started today. First of all, let me start by thanking the various organizers from NIT Calicut for giving me this opportunity. First of all, I'd like to thank the head of the department, Professor Sujit, and the coordinators, Professors Anuj and Lakshmi, for doing the same, and the entire team of organizing committee who have made this possible. And most importantly, to the chair, Professor Shankarlingam, to have introduced me kindly. Thank you very much. And before we get started with this series, I would like to actually appreciate the effort by NIT Calicut in order to get students introduced to very advanced topics of NMR and mass spec. As a researcher myself, we look forward to students who have some exposure when they come for their PhD studies. And this definitely gives a great platform for all the students to learn, understand, appreciate what happens, and transition towards research in a quicker fashion. And in this aspect, I would also like to thank the organizers for having a wonderful list of speakers. I've been following as many talks as I could from yesterday. And the transition is also going to be easy for me, largely because I've been preceded by wonderful speakers across from yesterday until today. To start with, Professor Narayanan Kurur gave a fantastic, lucid presentation on introducing NMR, the pulse sequences, and trying to give an idea on chemical shifts, scalar couplings, and so on. And this was followed by Professor Raghotama and just now by Ravi Shankar a few minutes back on trying to give an idea on how two-dimensional NMR spectroscopy is very useful in order to adjudge a structure of peptides by Professor Raghotama and of natural products by Professor Ravi Shankar. Not to forget Professor Abani Buyan, who provided a good background on how NMR is useful towards understanding or even characterizing protein structures. I'm going to be changing a little bit of my gears here. I'm going to be talking about nucleic acids. So I'm going to be trying to introduce as much as possible. While my camera is there, I'm going to be looking at my tablet down here and speaking. So if I'm looking down most of the time, kindly don't mistake me. It's largely because I'm writing. I'm trying to write while I talk. As Professor Ravi Shankar also mentioned, please feel free to stop in the middle of the discussion. I'll try to pause in the middle. I have divided it into two sections where I try to give you some basics followed by an application, as you have been seeing for the past 2 days. Some basic concepts in NMR are introduced, followed by applications that they're trying to showcase in different types of molecules. So therefore, that's the same strategy I would be taking. So please feel free to stop me if I'm going a little too fast. Ask me what's going on. I'll be more than happy to discuss with you. All right, as the chair already introduced, I'm from Indian Institute of Science Education Research, Bhopal. And today I'll be talking about how NMR chemical shifts in particular would be helpful towards structural elucidation of DNA quadruplexes. And the results of this has been published last year in Journal of Physical Chemistry Letters. So people who might want to know more information are more than welcome to ask for a copy of the same. So let's get started on what is NMR and what do we look for things when we record an NMR spectrum. What you see on the right is a protein molecule by name bovine pancreatic trypsin inhibitor. This is a 55 amino acid protein. And basically, this is one of the very well-studied proteins using solution state NMR spectroscopy. Not sure how many of you are very comfortable with protein structure, but you don't have to worry about it. All that we are trying to say, so there are about 500 odd atoms that are present in this big molecule. And as you're able to see the tertiary structure, there are different types of tertiary structures that are present. There's an alpha helix. There are different beta sheets that are present. There's also a smaller helix called a 310 helix that is present. And as you might already know, proteins are made up. Once again, Professor Raghotama did give a nice introduction on what are oligonucleotides, what are polypeptides, and what are a protein. This is actually a protein which has a certain function in the cell. So it's a representation of an amino acid, which is characterized by an amino group and a carboxylic acid group, and also has a chiral C alpha group that has a H alpha that is associated with it, and the side chain group, which could be different for different amino acids that you're looking at. And when you end up forming a polymer, you would end up seeing something that you're able to see on the top right now, where this just concatenates with the amide bond, and then you actually form a polypeptide. So once you end up forming a polypeptide, if you try to record an NMR, generally people end up using water as the solvent. We have something like 90% to 10% water to D2O. And D2O is used as a field frequency lock in order to get a high resolution NMR spectrum. An example of a proton 1D spectrum is actually given here, where you're able to see the amide protons end up resonating downfield, and the H-alpha protons end up resonating slightly upfield, and all the other aliphatic protons end up coming even more upfield. You might always ask, oh, there are some protons that are coming even more upfield to 0 ppm. That comes up due to the methyl groups that are present in the core of the protein. And of course, there's a region here which I did not mention earlier, which is the aromatic region, which once again comes up due to aromatic amino acids such as tyrosine, tryptophan, and phenylalanine, and sometimes histidine as well. So what you are able to see is that, okay, depending upon the type of amino acid, we have a good enough resolution when it comes to what you see in 1D NMR. However, let's say for a 55 amino acid protein, you're not able to distinguish what kind of resonances come where. So this is an important aspect of NMR, where we would like to get as much as an atomistic characterization as possible so as to get a three-dimensional structure. So there are a few things that are associated the moment you start going to bigger systems, especially with solution state NMR. So multidimensional NMR is a necessity where, unfortunately, as you just saw, you're not going to have very resolved resonances. Let me take a quick example here. Sucrose is a small molecule which is of 342 Daltons, which is gram per mole. And what you're able to see here, sucrose has, if I remember right, C12H22O11. And if you slowly sit and count, you would be able to get as much of the resonance. I think it's not glucose, it's probably sucrose, which is C6H12O6. And if you calmly sit and determine, you'll be able to assign all the 12 protons that are present. And immediately what you're able to see is that you could get an atomistic characterization, which was once again very well explained in the previous lecture by Professor Ravi Shankar. But the moment you go to a large protein like BPTI, what ends up happening is that you start to observe two different things that happen. The number of protons increase tremendously, and you start losing the line width, meaning that every resonance is associated with a full width at half maxima, and you start going bigger in system, the full width at half maxima keep increasing, and due to which what you're able to see is that the resonances become broader and broader. Although for sucrose, you did see very narrow resonances, the moment you start going to larger biomolecules, this is a problem that starts to come up. So thankfully, there have been wonderful NMR techniques that have come up, which help people still get the information that they want. So I'm showing the same one-dimensional NMR spectrum here, but then what we start doing is to resolve the chemical shifts in 2 dimensions. So this is a 2-dimensional NMR spectrum, and this is what was introduced as a heteronuclear single quantum coherence spectroscopy in the previous lecture just a few minutes back. And what it does is that wherever you have a CH bond that is present, you're going to get a resonance. I'm just circling one of the examples here, but if you sit and slowly count for BPTI, you'll start counting about, so remember, this has all the C alphas here, the C betas would come here, the C deltas and the gammas that are present in different amino acids would end up coming a little more upfield. Of course, you're once again able to see upfield of 0 ppm, there are still some methyl groups that end up coming, which indicates it's a well-folded protein. Not just when it comes to carbon proton 2D, one can also use the amide proton. Remember, it's bonded to a H alpha, the C alpha is also having a nitrogen that it's attached. And you can also perform a single bond correlation spectroscopy between the nitrogen and the proton, which is called the fingerprint of the protein molecule. And what you're able to see here, this is a nitrogen proton HSQC spectrum of BPTI. So the moment you're able to understand this, you're able to appreciate the fact that, okay, from the 1D NMR spectrum, which has poor resolution, the moment you do 2D, you can start picking every atom specifically. This is a very important aspect. And for people who have done NMR spectroscopy to determine whether they have successfully done their organic molecule synthesis, one has to remember this is different from biomolecular NMR, because in biomolecular NMR, we ensure the molecule that you're studying is extremely pure. Similar to the example that Dr. Ravi Shankar did show a few moments earlier, where the natural product that he is trying to study would be at utmost of purity. But on the that's the same thing that we end up doing for biomolecular NMR as well, where the protein or the system that you're trying to study will be as pure as one could get. So this slide quickly gives you an idea of different amino acids that are present. And once again, what I'm trying to impress you upon here is that the carbon-13 chemical shift, which is being shown on the right side of the panel, depending upon the amino acid, I'm just picking alanine as an example, and then trying to show where the alpha and the beta carbons end up coming. On the other hand, if you just take a similar amino acid, let me take phenylalanine, which just quite different from one another. So therefore, what you are able to appreciate is that depending upon the side chain R group, the chemical shifts are going to be very specific. And we also saw fantastic examples yesterday elucidated in terms of peptides, where using a toxi, one could actually pick up a fingerprint based on what type of amino acid is present. A proton-based chemical shift measurement. I'm not sure whether it's getting updated. Yeah, so basically protons also are very specific. So therefore, when you actually acquire a two-dimensional NMR spectrum, one would be able to get as much information as much possible in from a given system. So why are we obsessing with chemical shifts? I'm trying to jump to the topic that I want to discuss today. Why are we so much obsessed with chemical shifts? Is that if you're able to just get the carbon-nitrogen chemical shifts, it has been shown for a very long time, more than a decade, that these are very sensitive reporters of tertiary structure that are present in proteins. Nowadays, they have also taken to an extent where if one ends up giving the C alpha, C beta carbon shifts and the carbonyl carbon in association with the H alpha and the amino proton and the nitrogen chemical shifts, one can actually give it to a program called TALOS, which is what is shown here. It ends up running an algorithm and it ends up predicting the Ramachandran 5-psi angles for the polypeptide or the protein that you're trying to look at. And it actually gives you an output like this, where if you have a red region, it indicates, oops, I'm sorry. If you have a red region, it indicates an alpha helix. And whenever you have cyan regions, it's trying to indicate there's a beta sheet. And this has been extensively used in a de novo structure determination nowadays, where if you give chemical shifts, one can actually get a tertiary structure of proteins. You might ask, why is this so important? Because largely people are looking to get this tertiary structure in order to target a given protein system for different diseases. For instance, in a given cancer, a protein gets upregulated and you want to inhibit this protein, getting the structure would be extremely useful for drug discovery chemists so that they can come up with different systems. I'm sure in the last 1.5 years, you have seen an explosion of research in drug repurposing for the case of coronavirus. And this is all feasible just because you have some protein structures, nucleic acid structures that are already present in the protein data bank. And what NMR is able to achieve it is to get it in solution conditions and in a much, much faster way using chemical shifts. So let me now quickly switch gear to nucleic acids. I'll be talking more about DNA. Although people might have been introduced about it, I'll quickly give you a brief description of their chemical structure and tertiary structure in this slide. What you're able to see here, the polymer of nucleic acid is actually nucleotides that are covalently bonded to each other by a phosphodiester bond, which means position is actually switched to a hydroxyl group. And this sugar is in turn attached to a base, which is of 4 types, as shown in the right-hand side here in the B subplot, where you're able to see adenine pairs with thymine or uracil, but in a DNA, generally it's thymine, although there are some cases where uracil is also observed. On the other hand, a guanosine ends up pairing with a cytosine and with 3 hydrogen bonds. On the other is quite sensitive to protons. All that we will be observing here Let me try to erase what I've done. What you'll be trying to observe is the sugar protons that one can actually get with respect to their carbon correlation. Remember the one-dimensional proton-carbon correlation. And in addition, even the bases have CH protons that are present. So once again, going back to aliphatic and aromatic protons that are present. And in addition, you also have nitrogen proton correlations that one could get. And importantly, if you pay close attention, the nitrogen proton correlation gives you an idea of the base pair formation in DNA. On the other hand, the carbon proton correlations help you understand what kind of sugar conformations and base structures are present. The important thing that I'm stressing about sugar here is that a 5-membered sugar furanose ring could actually pucker in 2 different ways. And it could pucker in multiple different ways. And the 2 specific ones I'm showing here are the C2 prime endo, the C3 prime endo, where in the B form DNA, you have a C2 prime endo pucker, while from A form DNA, you have a C3 prime endo pucker. Might ask, okay, why would the pucker end up changing the overall structure, which is shown in the schematics here. If you take an idealized A versus B DNA form, depending upon the sugar pucker and the base rise that is present between base pairs, you would soon understand and appreciate the fact how much the structure changes. And an extreme example of this is a Z form DNA, where once again the Watson and Crick base pairs, as shown in the B subplot, is still held good. But then you're able to appreciate the fact how the backbone and the base pairs are quite different from one another. Just to quickly give you an idea of how the spectrum looks, this is an example of a system called Dickerson-Drew dodecamer. Dodecamer means it's a 12 base pair DNA, and it's a palindromic sequence, meaning that if you closely pay attention, the sequences that end up coming in the directionality of 5' and 3' are same in the both strands. So therefore, there's a C2 prime, C that are hydrogen bonded, the most downfield between something like 12 to 14 ppm. Unfortunately, it's not getting updated. Let me wait for a moment. So basically, the 3 protons that you end up seeing here correspond to a GC base pair. On the other hand, the downfield protons end up indicating an AT base pair that are present. Can one of the organizers please tell me whether the screen is getting updated?
Yes. It is updated.
Okay, good. So, and then you have a little upfield shift for the aromatic and the amino protons that are present. And as you start going a little further, these are the H1 prime protons. These are the sugar, as I was talking about a moment earlier, that end up being resonating at around, say, 5 to 6 ppm. What you see here is water. Water is suppressed here because water is present at about 5 orders of magnitude higher. The resonances that end up coming here are the H4 protons and the H5 prime and H5 double prime protons that end up coming. The H3 protons are lost in water, so I'm not indicating them here. The H2 prime and the H2 double prime protons end up resonating around 2 to 3 ppm. On the other hand, the methyl protons that are present in the thymidine nucleobase end up resonating upfield between around 1.5 ppm. So this gives you an overall idea of how the proton NMR spectrum looks. Although this does seem very well resolved, unfortunately, as you go to larger and larger systems, this resolution is not what is observed. The interesting thing that one also ends up getting is that as you keep changing the type of base pair, the chemical shifts are very sensitive. We are very comfortable with the AT and GC Watson and Crick base pair, which was proposed back by Francis Crick and James Watson back in 1953. But what was actually observed in 1959 is that the AT base pairs could also form a Hookstein type base pair where the glycosyl dihedral angle, the chi angle, if it's flipped by 180 degrees, what you're able to see is that AT still forms 2 hydrogen bonds and a stable base pair. On the other hand, GC requires a protonation of the cytosine to form. Nevertheless, this flip of the base still stably forms a AT and a GC plus base pair. And the carbon chemical shifts, it was observed back in 2011, are extremely sensitive for such a base pair formation. And I'm going to illustrate just in the context of a duplex DNA, how these chemical shifts are very useful. And in this aspect, I'm just going to be taking 2 model systems, one which is called as the A2 system, the other what is called as the A6 system. And I'm showing the same thing here. What are the differences between the Hookstein and the Watson and Crick base pair? And as you're able to see, the moment you end up changing side chain dihedral angle from anti to syn, you end up forming a Hookstein base pair. Although you might ask, okay, well, this is a very small change. Why would it cause any difference? As I had shown to you a little moment earlier, small changes in sugar pucker ends up changing the tertiary structure altogether. And the basic premise in biology is that structure and dynamics drives function. Although there are exceptions to this rule, but what I'm trying to make you understand is that subtle changes could end up resulting in markedly different functional rules of these biomolecules. And I'm just trying to illustrate that with one example here, where we took a system, we just ended up changing one given base pair with a methyl adenine. And a methyl adenine ends up having steric clash if it has to form a Watson and Crick base pair, due to which the chi angle switches from an anti to syn so as to form a Hookstein base pair. And the example also illustrated with the help of a proton 1D immunospectrum. And what you're able to see, most of the resonances almost stay wherever they are. There are subtle differences that come. But most importantly, the base pair, which is 9th base pair, which ends up getting perturbed due to this Hookstein formation, clearly shows a huge chemical shift difference that ends up coming. As always, the 1D NMR spectrum is good, but then once you're able to get 2D NMR spectrum, you actually get additional information based on the chemical shift perturbations. And this example, what I'm trying to show you is the chemical shift difference that comes up for the nitrogen that's present in the thymine base between a Watson and Crick, which is shown in red, versus a Hookstein that's shown in blue. And this change is done only in a single base pair. And what you are able to see, very, very small change in the base pair ends up changing the nitrogen chemical shift and the proton chemical shift upfield, which is known to reduce the strength of these base pairs. So basically, base pairs end up interacting with one another. The bases end up having dynamics, and due to the weakness of the base pair, one ends up flipping out more often than not, which could have a functional context by itself. And it is just not seen in the sugar, in the base pair formation, but one also observes differences that comes in the sugar. If you might remember, let me quickly draw the sugar structure as well, where this is the H1 prime proton and the C1 prime. I'm trying to show you the carbon proton HSQC, and what you're immediately able to see, the 16th position, which is modified, clearly shows a difference that comes in chemical shift. As I've shown you earlier, the moment you form a Hookstein base pair, chemical shifts of the carbon go downfield, and that's the exact same thing we are observing. Although this is known information, there are a few more things we started to observe here. So let me try to point a few important things that we ended up observing. One important observation is that the nucleotides that are close to this modification, remember G10 is also close to this base pair, and also T8 is close to the base pair that you are having. And then where is A17? There you go. The A17 also getting modified. And the most important aspect, in addition to chemical shift perturbations, you're also able to see that the carbon line width increases a lot. As you might have remembered from Professor Raghuttama's discussion yesterday, if you have very sharp resonances, that indicates a single structure is present. And the mere fact that you start seeing very broad resonances indicates other states are sampled, meaning that the given base pair is actually not very stable, and it's actually seeing many different conformational states that are present. All right. So that's important information that one is able to get from the 2D right away. And it just does not stop there. Even when you start to get the aromatic carbon chemical shift, you start to see similar kind of broadening. I don't want to go too much into the details of it. These are all published again. You might take the liberty to go take a look at it. But overall, what I'm trying to say is that these results, once again, complement the conventional NOE-based data analysis, distance change of about 4 angstrom to 2.5 angstroms, which results in a change of resonance intensity about, say, 1 to about at least 4 to 10 times more intense. Since the distance reduces, since it goes 1 over r⁻⁶, a anti to syn change is very well observed in the NOESY spectrum. But then, if you're able to take a look at the carbon broadening, you're able to see additional modes that are present for dynamics that is present. Once again, you can also see the same ring flip that comes from NOESY experiment from the H2 prime to the aromatic base as well. So I'm not going to go much into the detail because the current discussion is more on chemical shifts. So to quickly wrap up this story, what we were able to study is that a single base pair substitution, this, by the way, this modification that's present in adenine is also known to be a damage modification. A damage modification is one where a cell is not very happy if such a change comes up. And this is important because when people end up smoking, the adenine ends up getting modified to ethanoadenine, which also end up sampling Hoopstein base pair. So what we were able to show is that the gray is the idealized A form and the red one is the control system, while the purple one is the modified system. What we were able to see is that as you keep ending up forming the Hoopstein base pair, there's more and more DNA bending that ends up happening, which has different functional contexts. So now I'm jumping to the last part of this discussion where I'm going to be trying to illustrate how carbon chemical shifts could be useful to get an idea of structures of G complexes which have their own set of complications. I'd like to pass here if there are any questions that may come up from audience. If I grab a little bit of water. Any questions? Okay, so let me quickly jump into a specific type of DNA, which is called DNA G quadruplexes. And the G quadruplexes are formed by G tetrads, and the G tetrads are actually formed by a guanosine base. It should be noted that in a guanosine base, there are 2 hydrogen bond donors and 2 hydrogen bond acceptors. What this results in is that beautifully the G's can pair with
Hello,
one
sir.
another. Yes,
Sorry to interrupt. Your voice is breaking.
I see. Just one moment. Maybe I can try to change my. Probably due to an internet connection. Is it breaking all the time or it just started to break?
Just this session it was started breaking.
It just started breaking, so it could be an internet issue. Let me try to do something. Let me try to switch my mic and please let me know whether it's okay.
Okay.
Is my audio better?
Yes.
But is there a feedback or something?
It is clear now. You may please go ahead.
I see. Just a moment. Is it better now?
Yes. It is better.
Hello? Yeah, I hope it's audible now.
Previously it was fine, but now I feel a little bit echo is there.
I just one moment. This is the problem that ends up coming at times. Just one moment. Is the feedback gone? Is still there?
So still I feel echo is there. So
I see. Just one moment.
Yeah.
I think now the echo should be gone. Is that right? Is the echo gone?
Yes. Please go ahead. So you are not audible. You are not audible.
Oh, I'm sorry. Somehow I got muted. Yeah, is it audible now?
Yes, you are audible.
And I hope there is no feedback.
Yeah. There is no echo. Yeah, you can go ahead. Yes.
Okay, good. So the advantage of a guanosine base is that you have a hydrogen bond donor and a hydrogen bond acceptor, which nicely forms a tetrad as shown here. But the interesting thing that happens once you form a tetrad is that the oxygen atoms are really close to each other, which have lone pairs, due to which they are not very happy. And
Sorry to interrupt you. Again,
Yes.
again some issues there.
I see. Let me see. I hope it got better now. Hello? Is it better?
Yes, sir. It is okay.
Yeah, I'm very, very sorry about this thing. This sometimes happens where since I'm using 2 different connections, at times it happens. But now I've completely disconnected the other one. If there is any confusion, please let me know. Anyway, now coming back, the advantage of G tetrads is that they can beautifully form an aromatic pseudoplane where one ends up donating the hydrogen bond, the other part ends up accepting the hydrogen bond. From here on, I'm going to be showing this as a part of cartoon where all these blue ones indicate a guanosine base. It could be blue or pink based on their side chain conformation. And let me quickly try to tell you why studying about G-quads are important, because they are functionally relevant in multiple biological contexts. For instance, they are actually formed in telomeric regions and are also formed in promoter regions of oncogenes. Oncogenes are cancer-causing genes. And there are places where if these end up forming, the protein that's present or the gene that's present downstream for the protein to be transcribed would end up being expressed, causing cancer. So scientists are very interested to get the structure of G quadruplexes so that they can be targeted drugs. So therefore, getting the structure is very, very important. And where do we go from G tetrads to G quadruplexes is that when multiple tetrads end up stacking on top of each other, then they form a G quadruplex. And the important thing here about G quadruplexes is that their backbones would have different topologies. What do I mean by that? It could have a parallel topology where all the 4 phosphate backbones that are present end up running 5' to 3' in a parallel fashion, which is called the parallel topology. On the other hand, you could have a hybrid 3+1 where 3 of them run parallel and 1 of them runs anti-parallel. So this is called a hybrid 3+1. On the other hand, you could have a 2+2 anti-parallel where 2 of the adjacent strands are parallel to each other, while a set of 2 strands are anti-parallel to one another. So this is called anti-parallel 2+2. Alternatively, you could have all the adjacent strands anti-parallel to one another, which forms a fully anti-parallel topology. Just to give you an idea of what are conformations, I'm sure you have learned it in your basic chemistry course, where if you're taking a butane molecule, you could have the methyl groups that are completely anti to one another. Of course, this is a staggered conformation, or you could have a case where they're eclipsed on top of one another. And you know for a fact that the staggered conformation is more stable than eclipsed conformation, and they are separated by an energy barrier of about 19 kilojoules per mole. And similar is the case for guanosines, where the side chain dihedral angle, the chi angle, which I did mention about the Hooke's base pair a moment earlier, is the same case here, where the anti versus a syn flip ends up causing a good amount of change that ends up coming. Although in this chemical structure, you might only think that it's a very small change that ends up coming, but in reality, there are larger changes that happen based on the proximity of the aromatic proton to that of the phosphate backbone that is present. So without going into much details, I'll quickly give you an idea how this ends up coming. So basically, the guanosine base could either be in the anti-conformation or the syn conformation. And from now on, if you see a blue color base, you have to imagine a guanosine that's an anti-conformation. Or if you see in the pink, you're going to imagine a syn conformation. And the adjacent backbone strands are actually connected by loops, which could be a propeller loop, which connects the adjacent strands in a parallel orientation. Or it could be in a lateral loop, which connects adjacent strands in an anti-parallel orientation. Alternatively, you could also have loops that end up connecting diagonal strands in an anti-parallel fashion, which is called a diagonal loop. I'm going to be coloring the propeller loop in red, the lateral loop in orange, and the diagonal loop in green as we go forward. Brace yourself for the next slide. This tells you how polymorphic G quadruplexes could be. What I'm showing in the left-hand side, not sure whether the slide is getting updated. What I'm showing in the left-hand side as a PPP, yeah, I think now it got updated. In PPP is that this is a parallel topology, G quadruplex, where all the 4 strands are parallel to one another. On the other hand, if all the loops are in lateral fashion, you are going to see you form a fully anti-parallel topology. Brace yourself for all possible combinations. If you're having some combinations of different forms of lateral, propeller, and diagonal, you could form 6 different forms of hybrid 3+1. Once again, if you pay close attention, 3 of them are parallel, 1 is anti-parallel. That's the case for all of the ones that are shown here. On the other hand, you could also form 5 different types of anti-parallel 2+2. This complicates the way in which one can get the structures. And for a given sequence, all this could be folded. I'll just give you one example of this case. If you're able to take a look at the sequences that have been studied, which are telomeric DNA repeats, the core of the DNA sequence is literally the same. Only subtle changes end up coming in the 5' terminal and the 3' terminal. And what you are able to see is that the topology is quite different from one another. You're able to see the 1KF1 structure is actually a parallel topology, while the 2JPZ is actually an LPL type 2+2 topology. On the other hand, the 143D is a hybrid 3+1. So the same sequence could end up folding into all of this, which makes the analysis quite complicated. Just to give you an idea of the spectrum that we end up measuring, you remember the duplex ended up giving immunoresonances between 12 to 14 ppm. The beautiful part of the quadruplexes is that they give immunoresonances between 10 to 12 ppm. When I mean the immunoresonances, the ones that end up forming the hydrogen bond. So these are very well resolved so that people can study. And an example is given here where we ended up using a NOSI-based approach to get a sequence-specific information of which guanosine resonates at which part of the NMR spectrum. Okay, so this is a proton NMR spectrum. Now, one can also end up getting a two-dimensional HSQC, as I mentioned several times before. This gives you a much superior resolution. And using the NOSI experiment, we were specifically able to assign each guanosine that is present, and you're able to see a beautiful chemical shift resolution that one ends up getting the aromatic C8 chemical shift, and also in the sugar C1 prime H1 prime that's shown on the right hand side. So all these chemical shifts are useful, but we wanted to ask, can we actually just use chemical shifts and then solve the problem of the polymorphism that one ends up seeing in G quadruplexes? So basically all I want to say, from chemical shifts, can I actually say what type of topology ends up forming? I'll come back to this slide in a moment. So when we paid close attention to different topologies that are present, and when we looked at the biophysics behind it, what we were able to understand is that the parallel topology could only have medium grooves that are present. Due to dearth of time, I'm not going into the details of the biophysics. And what we were able to realize, only three of these planes, three of the blue planes can stack on top of each other, or three of the pink planes can stack on top of each other, or any of their combinations. Meaning, you can have a three planes of blue, or three planes of pink, or 2:1, or let's say 0 to 3 here, 3 to 0, and 1:2. So all this restricted the total number of blue to pink that one ends up seeing. Meaning that for parallel topology, it's going to be 12 anti to 0 syn, or 8 anti to 4 syn, or 4 anti to 8 syn, or 0 anti to 12 syn. So these are the only possible ratios we predicted based on the biophysics. Similarly for the hybrid 3+1, what we were able to see is that a 9:3, 7:5, 5:7, and a 3:9 are feasible. What I mean by this ratio is an anti to syn ratio that ends up coming. Yeah. So once again, let me go back to the more complicated slide that we showed here. If you pay close attention, you will be able to see these ratios of pink to blue seem to be maintained. So we got very excited, similar to the duplex DNA, where Hookstein was able to distinguish between the flip of the base. We asked whether chemical shifts are useful probes to get this. So before going there, let me try to quickly give you how do people end up solving these things traditionally. Back in 2012, people were able to show that either using the NOSI walk, one can actually get this information. But the problem is it's very laborious, meaning that you got to check across every possible topology, which makes life difficult. Alternatively, people could specifically label some of the guanosines with 15N or 13C or 2H so as to get multiple samples to get their assignments with different samples. The problem with this is that these are very expensive and require multiple samples to be prepared. Alternatively, people are also published long-range carbon-proton coupling experiments which help them get the same type of information. But the problem is that when you have less resonance dispersion, this will create a problem. And also these experiments are very less sensitive, requiring about 2 to 3 days of data measurement, which is too long. So what we were able to do is to take our hypothesis and check across a structure survey from the PDB. We mined for data from the PDB and asked whether our ratios are correct. And to our surprise and also excitement, we were able to see the parallel was indeed formed in a majority in the ratios that we predicted. Same for the hybrid and the anti-parallel topologies. There were ended up coming, as again, due to dearth of time, I'm not going to be discussing that. So overall, what we were able to see is that the parallel topology did come up with the even ratios of 12 to 0 and 8 to 4, and the hybrid ended up coming with a 7 to 5 or 9 to 3. The anti-parallel always came up with a 6 to 6. So the next question was, can proton chemical shifts be useful to determine between the blue and the pink? And unfortunately, except for one of the protons, all other protons ended up stacking on top of each other, indicating that proton chemical shifts aren't useful. Once again, we went to the BMRB to ask whether there is any data that is present to say syn versus anti-bases can be distinguished. Although we did have some data to say that the anti and the syn were actually well dispersed, the problem came from the fact that all of the data that has been deposited have some sort of modifications that are present in them. Remember, NMR and chemical shifts are very sensitive parameters. The moment you have any modifications that are present, it might end up changing the chemical shift. So we did not know whether the syn changes came up due to the structural change or due to the modification that is present. So we ended up preparing a bunch of 18 different sequences, folded them across various topologies. Once again, all these details are present in supporting information on the paper that's available for all of you to take a look, which ended up forming different types of loop to fold into every person. Jumping to the results, what we were able to see is that the carbon chemical shifts are extremely sensitive to resolve syn and anti. So what do we mean by this? Coming very close to the conclusion, if you run a carbon proton HSQC, if you end up getting your aromatic carbon in this region, then you can say for 100% certainty it's actually an anti-ring. On the other hand, if you end up getting a chemical shift here, then it's a syn ring that is present. Of course, the data statistics also show the same. And therefore, one will be able to count the number of anti versus syn bases so as to make the judgment based on what kind of topology is present. If you count the number of anti-syn, you know the ratio. If you know the ratio, you know the backbone topology. In addition to the aromatic chemical shift, we are also able to get a C1 prime H1 prime chemical shift, which is sensitive to other parameters. As I said, due to dearth of time, I don't want to go into the detail of it, but we were able to get much more specific structural features that come from carbon chemical shift. So in order to conclude how chemical shifts could be useful, one can end up preparing a sample at natural isotopic abundance. You don't need any specific labeling. Run an NMR spectrum to make sure you get an immunospectrum, and then run a HSQC, and then try to see what kind of ratios you end up getting, and then you would be able to decide what type of topology ends up forming. So I hope I have illustrated to you how chemical shifts are super helpful to solve the degeneracy issues when it comes to G-quadruplex structures, which could be multifold. Of course, we went ahead with this. We wanted to ask, there is so much proton information, can we use it in some way or the other? As I said, without much further details, we were able to see that the chemical shift clustering for the proton-proton correlations were excellent based on the structural features we were able to see. And then we developed a machine learning algorithm which helped us categorize the chemical shift based on which planes they arise from. And once we ended up taking this up, what we did was to take all the chemical shift information, train a computer to use this chemical shift in order to predict something, and then we tested on the systems that we already have. And what we were able to see, once we optimized it, we were able to see that for parallel topology, we could predict with 99% accuracy. If you give unassigned chemical shifts, we can actually say what type of structural features that they belong in. Basically, whether they belong in plane 1, plane 2, or plane 3, followed by which we could use a NOESY spectrum for analysis. Same thing for the hybrid 3+1, so I'm not repeating it. So we wanted to test whether this protocol would work. Remember, science is all about coming up with a hypothesis and testing it. So we came up with the hypothesis that chemical shifts could help in alleviating the structural degeneracy or polymorphism of G-quadruplexes, and we were able to show that the carbon chemical shifts could distinguish between syn and anti. And now what we were wanting to ask is, okay, does our method work? We took a sequence which folds into two different topologies in the potassium and the sodium condition. And as you're able to see, the chemical shifts are different, indicating that these form two different topologies. When we acquired the carbon proton spectrum, we were able to see in the potassium state, all the 12 resonances ended up coming in the anti-region. On the other hand, for the sodium state, 7 ended up coming in the anti-position and 5 ended up coming in the syn region. So therefore, it's a 7:5, and we were able to say that in addition to the C1 prime H1 prime chemical shift, these end up forming a parallel topology, the potassium state, while it forms a hybrid 3+1 in a sodium state. Once again, we were able to confirm this with the NOESY experiment, which is the traditional way, and we were able to get the first initial of chemical shifts in order to get DNA nucleic acid structure using this methodology. So overall, this is where I would like to stop. is that you saw how chemicals using a computer and machine learning algorithm will also be able to get some tentative assignments with which you would be able to reduce the total amount of time required to analyze the systems from several weeks to hardly a day. So with this, I would like to stop my presentation. So basically what I was able to show is that using NMR as your torchlight, you could pick up different topologies that gives you very specific heteronuclear correlations spectrum. Therefore, one can actually come up with specific topologies and assign them. And all of this wouldn't have been possible without the generous funding that I've gotten from SERB and ISA Gopal. I'd like to thank SERB here again for funding this wonderful workshop where students are enlightened on learning how NMR could be useful towards multiple applications, including natural products, peptides, proteins, and small molecules. Not just NMR, of course, also HRMS, both of which are very complementary techniques. And before I conclude, I'd like to thank the people who worked on it. These are my lab pictures from different times. This is from 17, this is from 20, and this is one of the latest pictures from 21, which we got about a few days back. And this work was done by Rajesh, who is a PhD student, and Manish, who's also here, and by Anup. So without these people, this wouldn't have been feasible. And if you guys would like to know more information, I'd be more than happy to take questions, or the papers have been already published. So please do take a look. Thank you very much for the wonderful opportunity. I hope people learned a little bit of NMR and biomolecules from this lecture. Thank you.
So now the session is open for the discussion. So participants, so in case if you have any queries, so you may please raise your hand and our team will unmute and you can ask directly the question to Dr. Bharatwaj. If you are not able to do that, you can chat your question in the chat box. So Dr. Bharatwaj would be happy to answer.
Totally, that's correct. Feel free, don't be inhibited. The whole point of this exercise is for you guys to understand a little bit. And I hope I have done a little bit of justice in trying to keep it as basic as I could so that everybody could benefit. So feel free to ask questions. It doesn't have to be students, even the faculty, PhD scholars who are around, if they would like to ask questions, I'll be more than happy.
So participants, you may please ask your questions. So in case if you have any doubts. Okay, so seems there are no question about it. So, but I have only one question, Dr. Bharatwaj.
Please go ahead.
As I am a bioinorganic chemist person, so and I try to correlate what you are doing with me, closer to my research. So you made a lot of peptides, right?
No, these are nucleotides, oligonucleotides, but similar to
nucleotides, right?
Yeah.
So how about putting a metal inside and then checking the?
That's a great question. People love to do this. Actually, we have started the same thing. We have started actually a bioinorganic project just this summer with us master student. As you might remember, the tetrads are really close to each other. There are lone pairs of oxygen that end up repelling from one another. And this is indeed stabilized by multiple, many different cations. The most common ones are potassium and sodium, but also strontium, magnesium, lanthanides, transition metals end up stabilizing this. So I can answer your question in two different ways. People have looked at how transition metals end up stabilizing these tetrads as well. That's a definite area of research where people are really interested in asking questions that you are thinking about. If I do put an iron or a copper, can I actually catalyze something and make things cheaper? For instance, this could be towards something which is not otherwise feasible. You can put it in water and still have the catalysis going. The main problem of iron is that it's not very soluble in its catalytic state. Iron 3 is less soluble in water. So if you can actually coordinate with the quadruplex, it helps. So people have tried that. On the other hand, what people have also tried is to actually get inorganic complexes, which ends up stacking on the quadruplex and then initiating catalysis by themselves. So that is something that we are trying to look at. There is a paper that is published in ACS Catalysis earlier this year, where they were able to show cyclopropanation that has been facilitated in an NHO selective way with the help of porphyrin, iron porphyrin ligand or system, iron porphyrin complex that's stacked on top of a quadruplex. Yeah, so that is something that we are looking into on how these quadruplexes could help in bio and organic chemistry. So that's something that we are looking at very actively.
It's very interesting to understand this actually. So
totally, yeah.
Because I'm in my group also, we are working on a lot of metal complexes and trying to perform some catalytic reactions. So
I see. Okay, that's
so
great.
there is
So
one.
Yes, there's a question from Shivangi. How
Yes.
determination of genetic anomalies by NMR is done? It's a great question. So first of all, NMR does not do genetic analysis. On the other hand, NMR could try to understand if the sequences are different in a very, very subtle, in a very small way. How would things work out? That's actually what we are working on. I'm going to be giving another NMR workshop next Tuesday. Sorry for the advertisement. I'm actually going to give on how last 50 years of NMR has contributed towards understanding DNA structure using various methods. And in that aspect, I would like to point out to you, many people try to think only proteins have diverse structures. This is actually not true. Proteins have diverse structure largely also because they have multiple types of amino acids that are present, 20 different types, which are common. On the other hand, nucleic acids just have 4 types, A, T, G, C, and they can only pair A with T and G with C. People think, okay, this is very trivial. That is actually not true. If you think about it, the gene that I have and you have, or anybody who's listening to it right now, have is literally the same. In fact, the genes are, the chromosomes share about 98% similarity between us and chimpanzees. But we are not chimpanzees. Although you and I have 99.99% of the genes literally same, you and I don't look similar. We don't talk similar. We don't behave similarly. So what people have come to understand is that subtle differences in the DNA sequence ends up changing the structure of the DNA. Therefore, different proteins end up interacting with them and silencing some genes versus activating others. Okay, so this is an extremely important aspect of where NMR could contribute. And in this aspect, we are once again currently working on it with a PhD student, trying to understand, can we come up with nearest neighbor models to say how different sequence-specific DNA signatures are. So although we cannot comment on genetic anomalies from NMR itself, but if the sequences of the genetic anomalies are known, NMR could come up and say why such anomalies come up. Okay, and this has happened for multiple different scenarios. For instance, some promoter regions would end up having a unique structure within the duplex itself. Maybe it ends up forming a Z form. Maybe it ends up forming a G quadruplex. So those things end up altering the entire way biology ends up working. And for that, structural biophysics of DNA with NMR is extremely important. Yeah. So there is another question. I hope I answered your question, Shivangi. If you have any further questions, please let me know. Kamini Patil has asked a question: how metal complexes fixed in pigments influence of plant pigments and make stable metal complex pigments? This is a question you should probably ask Professor Shankar Lingam here. I'm not an expert of how metal complexes end up stabilizing. On the other hand, how I could say this is that from a physical chemistry perspective, it's always trade-off between enthalpy versus entropic contributions that end up stabilizing something to drive it towards a delta G naught that's less than 0 to stabilize a few things. But all this would be determined largely based on what kind of interactions come up from the metal with the side chain of the coordination chemist, because at least there is 100 years of this thing. I think Werner came up with this hypothesis back in 1920, literally 100 years back. So I'm sure there's a lot of literature that is present on how metal complexes are stabilized in pigments, which probably ends up stacking. The pigments could once again be aromatic, so they end up stacking on top of each other and all that, which could be similar to what I ended up showing for a quadruplex. Unfortunately, there's not much commonality between what I gave and pigments, so therefore I cannot answer any further. I hope this helps a little bit. Yeah, so I'm happy that some questions are coming up. Feel free to ask. Don't be inhibited. It's your knowledge that you have to learn. The professors in NIT Calicut have organized it so that everybody benefits. And unfortunately, we couldn't meet in person. Otherwise, it would have been even more nice to share and discuss in person. And I should also thank the organizers because you guys ended up socializing some of the people whom I know for a very long time. Like to name them out, Professor Dinesh is senior of mine from IIT Madras, and so is Christy George. Christy and I work together. Christy will be talking in one of the later sessions. And also Professor Dilip Kumar Chand, who was my inorganic chemistry teacher. So Professor Shankarlingam, he might be a better person to answer these questions with respect to quadruplex.
Yes.
He might answer it from a mass spec perspective. So, but I hope it gives you something complimentary. So I wish we were in person, in which case I could have met all of these people over this week, but unfortunate that we have to meet in these circumstances. But I hope I get an opportunity to visit Calicut at some point in future.
Definitely. We will be inviting you in future. Definitely.
Thank you very much, Professor Shankarlingam. If there are no further questions, we can stop right here. Thank you.
Yes. So if there are no further questions, so let me join in thanking the speaker, Dr. Bharatwaj, for his excellent talk on biomolecules. So thank you. Thank you, Doctor.
Thank you very much. Have a good
Sure,
day.
we will meet some other occasions. We will invite you.
I'll be both and happy. Thank you.
Thank you, sir, for the wonderful lecture. Now it's time for lunch break. I request all participants to resume here at 1:45 p.m. sharp for the next session, NMR data analysis. Please be online on time. Thank you.
ചെയ്തു ടാബല്ല അയാളെ സ്ക്രീനിലുണ്ട് ഇതല്ലേ കാണുക
Yeah.
See here. Next is the swim point
ഒരു ഫീബിൾ അതിന് തുടക്കത്തിലൊക്കെ
spectrum. So the outline of this video include this video is presented to you by Methodology Lab.
കേൾക്കുന്നുണ്ടോ?
This is a tutorial for simple proton and NMR spectrum recording. We shall not discuss here too much of technical details.
Hello, sir.
Spectrum. So the outline of this video includes the sample preparation, how a simple and sample and get ready to insert in an NMR spectrometer. Next thing is the basic introduction to the NO machine software, which is used as an interface for different operation in NMR machine. How does it look like? Operate it. And finally, we spectrum recording, how to get a proton and spectrum and export it for further use. Again, this is just instructive video and talk about a specific Bruker and machines and software. So now the question is, proceed about recording the spectrum. So let's go. So for sample preparation, there are a few things required. This is here a sample vial of 1.5 ml. Can be used for mixing the solvent and solute. Instead of this, you can also use a simple glass foil. This is called the spinner. Stability to NMR sample. This is that it helps the
Hello, Vikrant. Can you hear me? Vikrant? Hello, Nagarjuna. Yeah, 1 second, I'll unmute you. I think you have to quit and join one time. You quit one time and rejoin. Yeah, now you unmute your mic. Okay. No, you are not audible actually. I'll call you.
Sir, am I audible? Sir, am I audible?
Yes, you are audible. You are audible. Yes.
Yes, sir.
Yeah, and control room, you can give the presentation right to Nagarjuna.
Hello, sir.
Yeah, you are audible.
Okay, sir.
Welcome back to second session of online workshop on methods of data acquisition and analysis in NMR and HRMS. We are going to start the NMR data analysis session by Dr. K. Nagarjuna from Advanced Center of Research in High Energy Materials, University of Hyderabad. I humbly invite Dr. Anujay Varghese, Assistant Professor, Department of Chemistry, NITC, to chair the session. Welcome, sir.
Thank you, Anshri. So this session is handled by Dr. K. Nagarjuna. He did his MSc from IIT Delhi, and after that he joined University of Hyderabad, and he completed the PhD in 2016. And since then, he has been working as a postdoctoral fellow at Advanced Center of Research in High Energy Materials. And his major area of research, as the title itself says, it is about the synthesis and characterization of energetic materials. Especially strained molecules are his area of interest. So this session is mainly meant for introducing this data collection, and as well as the sample preparation, and as well as how to interpret the basic NMR data. So this is not of very highly advanced level. So this is basically those who are started using this facility, or those who are interested in knowing how to use this facility and how to interpret that data. So for this, that's the aim of this particular session. So I hope that the students, especially the PG students, MSc students, and the research scholars in their initial years would be getting benefited by this one. With this, I request Dr. Nagarjuna to take the session, start the session.
Thank you so much, Dr. Anuj Varghese and Dr. Lakshmi Madam. It's very nice to know about the importance of NMR. So today I'm going to mainly talk about analysis part. Dr. Anuj Varghese, my screen is visible?
Yeah, it's visible. It's visible.
Yeah. Okay. So myself, Nagarjuna, in Abraham. So today we are going to do NMR data analysis. So before that, already everyone is listening important talks on NMR. So I don't want to give any brief detailed introduction, but all we know, nuclear magnetic resonance spectroscopy is a powerful tool in electrical chemistry and condensed matter science, as well as for the elucidation of structure and dynamic macromolecules. We know all nuclei possess charge and mass. Those with either an odd mass or odd mass number or odd atomic number possess magnetic spin. So for F19 is all NMR active. Even nuclei containing a number both protons and neutrons have equal to 0 cannot act in NMR. So we also know a nucleus without spin cannot be detected by NMR. So before taking our samples to record in NMR, we need to know the importance of the solvent, saturated solvents, because we most of the in our chemistry we take samples and dissolve in the solvent and we'll record the NMR. So because why deuterium has a even though it has a nuclear spin, but in general deuterium NMR absorptions are not detected under the conditions used for proton NMR. Deuterium does not couple with proton and it's effectively silent in proton NMR. And also it's called as a non-magnetic nuclei. Solvents are needed for solid and viscous liquid samples, because in the usual type of proton experiment, the sample must be in free flowing liquid state. We may think that why we want to only we need to select CDCl3 or like prepared solvents we can go without hydrogen containing solvents, like for example, carbon tetrachloride, the useful solvent because it has no protons and therefore no proton NMR absorption also. But however, many organic compounds are not dissolved in this solvent. And also, if the solvent contains protons, the mixing of the signal due to samples with that of solvent will occur. It leads to complex spectrum with the gained signals. Modern NMR spectrometers measures the deuterium absorptions of the solvent to stabilize the magnetic field strength by controlling the locks and the shimming of the shimming. So in general, the deuterated solvents mainly the routine in organic chemistry like CDCl and DMSOD6 and deuterated methanol and C3CN and S1D6 and D2O. But mostly used solvent in organic chemistry, mainly CDCl, because all CH based compounds soluble in CDCl3. And next commonly used solvent is DMSOD6, but preferably it's preferable for polar compounds. And the CDCl3 is because it's also cheaper price, and most of the compounds with record in CDCl3. And again, the choosing of solvent, it depends on entirely your nature of the compound. What type of compound you need to decide to choose the solvent. As for in general, most of the non-polar compounds, most of the CH and CH base are soluble in CDCl3, but most of the polar compounds soluble in DMSOD6 and S1D6. And especially the most of the nitrogen containing compounds, more nitrogen rich compounds, exclusively soluble in DMSOD6. And is a very polar solvent because it is like a CD3 S double bond O CD3. So that's a polar S double bond. It's like a polaric nature. So sometimes it may react with a molecule and it may cause to decompose the molecule. So in such case, we need to take other solvents like S1D6 and CD3CN, and these will be useful to record the NMR. And another D2O is very useful solvent for to identify in our compound whether OH or NH functionalities. It's a very useful solvent. For example, you have you know that your conversion you converted, for example, methoxy to OH. You need to know that it was my methoxy compound totally converted to OH. So sometimes we don't, because of moisture also we get some kind of peaks with OH. Then in such case, if you go for a D2O, then it will exchange and it will disappear the respective region. So like that, we can use D2O solvent. And these are the main solvents. Of course, there are a lot of other solvents are there, benzene, and we can use. It depends upon the your reaction and what kind of experiment you are doing. And second thing, in proton NMR data, these are the solvent peaks we can observe, the solvent residual peak. In CDCl3, we can see 7.26. What is the 7.26 means? Because in CDCl3, 99% CDCl3, but there is a trace amount of chloroform will be available, it will be present. So that's what you will get, that the CH of the chloroform around at 7.26. For example, if you see any spectrum, if you are having seeing the 7.26, that means this spectrum was recorded in CDCl3. And if you, in case of in DMS or D6, you can see 2.5. In case of CD3CN, you can see 1.94. In methanol D4, you can see 3.31. In case of S1D6, 2.05, and D2O 4.7. It's a these values may a little bit vary, but it in the range of definitely like 1 point. Suppose CDCl on 7.26 means definitely it will 7.25 to 26. And also another important used to observe in normal NMRs is the water peak. Like in CDCl3, 1.5 is the water peak, and DMSO 3.35, and in deuterated sonitrile 2.13, and deuterated methanol 4.87, and in S1D6 2.84. So about this particular set of solvents and what are other impurities in your molecule, we can go through this journal. This organometallics, it's a very excellent journal to know the chemical trace impurities, and they explain very nicely. So I given here the reference also. And another thing, in recently people are started using non-deuterated solvents also, like chloroform also used to record, but it depends on the what type of experiment we are conducting. However, there is no much data on not come on these non-treated solvents, because people are now thinking to replace treated solvents because because chloroform, for example, if you instead of CDCl3, chloroform is very cheaply solvent. So people are trying different techniques to use non-treated solvents also. And next, before going to record our sample in NMR, we need to prepare our sample. So this is the usual typical NMR tube with a cap, and then we have to take the respective amount of compound, and then solvent we have to take. So generally, we used to take 0.5 for any doing the experiment. And again, if you know, if it is depends, and see if we taking any CDCl3 samples, generally it required a less quantity for predicting proton and C13 NMR, because it is not a much polar solvent. For example, if you are taking 30 mg compound and CDCl3, you can finish within a 2 scans in proton NMR. But in DMS or D6, it will take some 16 scans to get the good spectrum. So like that, we have to, based on the solvent, we need to take the compound. Sometimes people used to take more, but that's in such cases, it is not good also taking more also. In general, if you're taking CDCl3, any compound, it's 40 mg is sufficient to get a proton number and C13. And in case of DMS, we need more amount of compound, because DMS is a very polar solvent. Other solvent S1, D6, this is also it is minimum amount is like how much you are taking series, that will be sufficient. So now we are going to listen the video on sample preparation. So Dr. Anuja, can you
Yes, yeah.
present the video?
So actually, if the students have anything to ask on whatever is told about the solvents and about how to choose the solvent, if they have any questions, they can ask. We can address it just before going to the. You can chat, type the questions in the chat window, or you can raise your hands. Okay. Yeah, I think there are no questions. So from the control room, can you share the, can you give me the presentation rights? So this video was shared by Professor Kurur, Narayan Kurur. So sir has handled yesterday's first session, in fact. So he was kind enough to share the recorded video. It was basically done by his lab at IIT Delhi. So we'll share that video. The audio is slightly low, so you need to listen it carefully.
This video is presented to you by Lab. This is a tutorial simple spectrum recording. Discuss their details. Spectrum. So the outline of this video includes the sample preparation, how simple and insert in a spectrometer. Next thing is the basic introduction to the NMR machine and software, which is used as an interface for different operation in NMR machine. How does it look like? How to operate it. Then finally, we'll learn about the spectrum recording, how one can get a proton NMR spectrum and export it for further use. Again, this is just an instructive video, and we'll talk about specific Bruker NMR machines and software. So now the question is, how do we proceed and how to go about recording the NMR spectrum? So let's go. So for sample preparation, there are few things required. This is here a sample vial of 1.5 ml. It can be used for mixing the solvent and solute. Instead of this, we can also use a simple glass vial. Now this here is called the spinner. It provides stability to NMR sample when it is inserted into the probe. Other feature of this is that it helps the sample if you need to rotate the sample. So there you can see this yellow, white, black strip, which is sensitive to magnetic field and helps in rotation of the sample. Now this is a NMR tube in which sample is placed, and this is inserted into the probe. This is a 5 millimeter diameter of NMR tube depending upon the diameters and the bore system that we have. Next thing is, after this is the measuring cylinder. It is used to check the level of samples in NMR tube. You see here, it has marks to measure the optimal length of the sample for various diameter of NMR tube. See here, this is 2.5, 3, 5, 8 and 10 millimeter. This checks the length of the sample, which also helps to properly lock the sample. So let's start the preparation of sample. We have made the desired amount here in this sample vial. We are going to use this micropipette to transfer the solvent and use rest of the things. This is the solvent we are going to use. It's a deuterated solvent. Here you can see deuterium oxide. And now we open the bottle and check here. This it is 100 microliter. Generally we use 500 to 600 microliter for a sample preparation. Transfer some amount to this. So now I have added 500 microliter sample. Let me close the bottle first. Close the vial, shake it to mix the sample. You have to make sure that the solvent that you are using is the solute is soluble in this. Let's check this. Now our solution is ready to transfer it to the NMR tube. Again, we are going to use the micropipette here to transfer it. For preparation of protein samples, and macromolecules, we have to worry about other factors also like pH and temperature, but this is a normal sample that we are trying to collect. Make it settle then. We are going to add the rest of it. Okay. All right, the sample is settled now and we are going to prepare it for the NMR machine. Take the spinner, put it over the measuring cylinder like this. Now insert your NMR tube like this and just check the length of the. Now here you can check that our sample level is above 5 millimeter marker. It should not be less than that or way too much from that. Take out the NMR tube, Just make sure you do not touch this magnetic strip. Ready. All right, so it's time for a walk around the spectrophotometer. This is how an NMR. There are some required accessories for this. This is a BCU unit for controlling the temperature in probe, and that there is a HPPR2 preamplifier, and here are the all the wires, and there in the center is a probe in which the sample is inserted. These here are the legs for support and to avoid unnecessary earth vibration. Now we are going to look at the upper part of this machine. So the upper part of this machine has a liquid nitrogen input. Here you can see this is a liquid nitrogen input. Those are the liquid helium ports. This here in the center is a bore in which the sample is inserted. That beside it is a nitrogen sensor which senses the nitrogen in the cylinder. That's it here for now. Let's go and take a look at other things that are required. This here you can see it's a console, the back side of the console which is connected to the spectrophotometer. This is here the computer or the operator console. Now this is the front part of the console. It consists all the electronics that is required for different operation, BSMS, IPSO unit, amplifiers, and the temperature unit. So now this is time for learning this software. The software which is used by Bruker is the top spin. You can see here the spin with showing a magnetic moment here. This is the top spin which is used as an user interface for console and the spectrophotometer and control all the operations. So let it open first. Let's see how does it look like. So this is the top spin window here. Close this. Now this is a commands for different operations here. So let me go through them one by one. Start, acquire, process, analysis, publish. Contain different sub commands. Here you can see this create data sheet for creating a data sheet, find data sheet to find a data sheet, open data sheet to open the already saved data sheet, paste data sheet from one place to another, and read the parameters required for the experiment. So next is the commands for acquiring the spectrum. These commands are used. Here you can see sample related commands are there if you want to eject the sample or insert the sample or other commands. And this here is the log command to log the sample. This command uses the tuning and matching. There are tuning and matching of the probe. So now this is the spin command which helps to rotate the sample in the probe. Shimming commands, there are various shimming commands here available. Prosol, it is used for setting the default parameters in any experiment. And this here is the gain to adjust the receiver gain. This is a go command for recording the spectrum. Now here are some process commands processing the spectrum, Fourier transformation, multiplication. This is for adjusting the phase. This is calibration, peaks picking, if you want to pick a certain peak, and this is the integration command. And then this is now the analyze subcommands which we use for different applications. And this is the publish command. We can print or export our data from here. So all these commands here you see, these commands can also be controlled in a different way. So by not using the above command, we can use this command line here and type our commands for directly recording the spectrum. Instead of clicking all these, we can just give some command to this spectrum and we can do the recording of the spectrum. So first of all, this here is the directory window in which you will be seeing your data which you have saved here, and you can see here different directories. So let me open one, and this you can see different folders for the experiments that you want to do. You can go here and add new directory here by using this command. Now once you open your data sheet by using this, you can see will be seen in this blank window here. Now below this command line there are small various windows. Here you can see the amplifier control. Once you acquire your spectrum, you will see a pulse here in different channels. This is the acquisition information. When you are recording the spectrum, you will see some information about your recording spectrum. This is the FID flash. If the FID is being recorded here, that can see in here. Now next is the lock. Here you can see 100%. There is already a sample in there, so it is showing 100%. And now this, whether the sample is inside the tube, the probe or not, you can see here. And next is the stream coil temperature. You can try this. Now this tells the probe temperature, and also it helps to control the temperature if you want to change the temperature. So if you click this, you will see a temperature unit which can be controlled here. You can see this tab has been opened. Next is the spooler. If you give multiple commands simultaneously, this will take care of that. You can see here. And the next is the BSMS status. Mostly it shows the automatic shimming that it performs, the data which shows, and next is the time. So all right, this you can see the lock that shows 100% here. This all can be controlled by, this also can be seen here by this lock display, or we can give the command here lock disp, and here you can see a window has been opened, but you don't see anything. It's a blank line because the lock is 100% though, so the signal is somewhere here, so you are not able to see that. So for that we will be using this BSMS control suite here, that is can also be opened here by a command BSMS disp. As you open here you can see this is a BSMS window. Here you can see the sample is locked here. And first of all, there are various tabs for different works, and you will not be requiring any of other tabs apart from this main tab here. You can see here. So this auto function automatically does the work for you if you are trying to do that. And this lock controls the different required things about the locking the sample, the power, phase, and gain of the system. And this is here for the sample. If you want to lift the sample, or let me just okay. So here you can see this is for the sample. If you want to lift the sample, spin the sample, or different different kind of operation that you may be requiring. Next thing is the shimming. This is for the shimming. You can control it by using this window here. These are the various shimming coils along different axes. And below here you can see the sample details. If it is down, missing or up, you can always check. I have gained the lock signal here, so you can see it is showing 60%, which is optimal. And here also you can see the amount lock. So what if we lock off? So if you are going to turn off the lock, here you will see this kind of signal. This is red, green, and this is nothing but the deuterium spectrum that is being recorded in the background. So now if we turn on the sample lift, so what will happen? Here you can see there is a question mark here and the sample is missing, and now it is up. So if you want to look at the machine, yeah, so this is how sample in the board looks like. You can insert it like this, or you can remove it or add the another sample, but I'm going to insert it again. So all this is controlled by the airflow that is provided by the pumps. When the pressure is high, the sample goes up. When the pressure is low, the sample goes down. So now if I turn off the lift, you can see the pressure is released and the sample is going down. So you can see here the sample is going down. Yeah, it has gone down completely. So you can see the deuterium spectrum that is also there again. Now here, yeah, so here you can see the sample is down, and now we are ready for recording the spectrum. So let me close this window. Yeah. So one thing that I can do, I can use any already recorded data and create a new data sheet from there. That is a little bit easy, but let me show you from the scratch how do we do that. So I'm going to type here EDC. So here, this is the datasheet window for creating the datasheet. Now here you can add your name, whatever name you want to add. So I'm going to put today's date here, 18 October 2020, NMR trial. Okay. So experiment number 1. Here you can provide the experiment that you want to do. If you are going to select, there are multiple number of experiments that you can do in NMR, but we are only worried about the proton spectrum right now. So I'm going to type proton. So here you can see proton. Set this item in the editor. You can see now the proton experiment is set. Now here you can add the solvent. This you can see I'm using DTO solvent, so you can always choose your solvent by this window. Get prosol, execute get prosol for your default parameters that will be taken. Now here, yes, so now here in the dir window, I am going to add the directory in which I want to add my data. So I'm going to use my directory here. Now we need to provide some title for the datasheet, NMR test. Let's yeah, so NMR test run. Now all set. Let's we didn't specify the datasheet. Yes, so here you can see the proc number. Here I have to give some number here to continue. Yeah. So now if you close your directory and open it again, you will see a new folder that has been created. 18 October 2020 NMR trial. Let me open this, and yes, so So this is the data sheet where we are going to work. These are the different tabs for controlling your different operations. This is the spectrum tab. You can see every no raw data available. And the next is processing parameters. You can set all the processing parameters here. They are different, which is generally set by the prosol command. Now this here is the acquisition parameter. So there are also number of acquisition for parameters, but we don't need to worry about all of them. We can go this here, the pulse like symbol. Click on this and you will see the only required parameters that we'll be needing for this experiment. So I have execute the get result, so all the parameters are already default here, so we don't need to worry about right now that. But now this is the title where you can set or change the title. This is the pulse program that we are using. It is a ZZ30 program in which we apply different commands. And now this is about the peaks because there is no data here, so it is showing like this. This is integrals again. This is the sample details if you want to add your details about your sample. This is for the structure if you want to provide some structure and compare it. So this is mainly used by this analyze structures and different commands. This is a little bit advanced. Now this here is the plot because we have not recorded anything, so it is showing error. So you can print this. You will be seeing the spectrum here. This is the FID for raw data available, and this is the acquisition. If the acquisition is running, you can see here. So now we are ready to proceed. Yeah, so let me give the log command. First of all, we have to log the sample. So give me the log command. So I can choose different solvents here. To choose here the D2O. Okay. Now here you can see the locking in progress. You can see whatever the status of your command. You can see just below the command line. So it is. So here you can see it is trying to log the sample. Once the sample is logged, you can see it is stable in a stable. Now here you can see the logging is done. After that, we have to proceed for the next command, which is ATMA. ATMA is automatic tuning matching adjustment. So it automatically tune and match your probe. So here again you can see ATMA in progress. So it is going to take some time. So tuning and matching basically is required to increase the sensitivity with lower power requirement. So here you can see the wobble curve. It is trying to, yes, it is trying to minimize it. Yeah, so it is done. Now ATMA is finished. The next task is to homogenize the field. For that we use top shim. Yes, so top shim is used for homogenize the magnetic field around the sample. So now here you can see that the log level has been turned off and the top shim has started. You can see it is recording some FIDs here, and after recording some FIDs, tries to optimize the line shape for better homogeneity in this in the sample, and it will take some time. Okay, so top shim has finished. Now you can see the log level is again stable here, but it is a little higher. So get the 60%. So this is what we see, that when higher the lock level, there will be better shimming, and you can see here it is showing around 72%. You can control the gain of this lock by using this auto gain command. You do this, you will see it has come back to 60% here. So now our next command will be receiver gain adjustment, RGA. It will record some FIDs to adjust the gain of the receiver. Here you can see the FID flash. So RGA is finished now. Now next is, just for the security sake, get ProSol. In case if the parameters are not set default, it will set the default parameters. So now get ProSol is finished, and now we can run the spectrum. So for running the spectrum, the command is ZZ. So once you run the ZZ command, now here you can see the acquisition information here, and now here you see this amplifier control. You can see the pulse applying to the 1H channel. So now this, here you can see the FID is being recorded. Yeah, so these are the number of scans that are currently running, and this is the name of the experiment about that. Now this is the FID signal. If you look a little closely, so yeah, so this is oscillating, and is oscillating decaying function. So that's what is a FID, or free induction decay. So let this recording be completed. Okay, so acquisition is finished now. Now we have to process our spectrum. For that we have to give the EFP command, exponentiation, Fourier transform, and phase. So it will show the Fourier transform of this FID here. Now here, if you look closely, so phase is not good. So for that you have to give the APK command, that is automatic phase correction. Now you can see the phase has been corrected, and for the baseline correction you have to use the ABS command, ABSN. So this will automatically align the baseline. Now your spectrum has been recorded here. Now you just have to export this. Before that, we have to also pick the peaks and also integrate them. So for that, we are going to the process command, process the spectrum that we have already done here. Now adjust the phase also we have done, and we don't need to calibrate the axis in this case. So for picking peaks, so if you see this, another window has been opened for peak picking. So we just 1 second, expand this. So now if I click this, should be to do. Generally we don't do it like that. Yeah. No, so we have to define the reason first. So, all right, so this is how, yeah, okay. So we have to define a reason for picking the peak. Generally we don't use this for peak picking. We just give some different command. I'll show you how we actually do it. So for that we have to give PP, peak picking, and here you can see it will automatically select the peaks from whole the spectrum. You can define the reason also, and minimum intensity which it will read. So I'm giving 0.02 here. Okay, so now here you can see it has picked all the peaks that are there in the spectrum. Okay, so the next task here will be integrating these peaks. So for that we have to go the integration command. Now again there is a different window here. You can see this here is the integration on and off. If it is yellow it is on, if it is not then it is on, off. So you can just select the region like this. You will be able to see the integration. First peak it automatically takes as 1 default, and for this reason this is a little crowded, so we have to turn off the integration. Now we are in expansion mode, and we can expand this spectrum, and now after expansion we can integrate this by clicking on the integration and OK. So we can integrate it just like before. Just complete this. Okay, so once this is completed, you can just save this by using this here. So save this. Now we just have to export this data to further processing. So how we are going to do that? We can just go to this plot, and you can see that your spectrum is visible. Below you can see the integration, and above you have to choose the label here. Okay, so now you can see the labels, all the labels, these meters here. Now to set a printer which you want to print, you can set according to. Okay, so you just have to now control P, and you can give the print command, but here I'm not going to do that. Just this is just for instruction. Now other way of exporting it is in the publish by using this PDF command, where you can use different PNG or different formats. Now for removing the sample you have to turn off the lock and again lift the sample. You can remove the sample and add another sample to continue like this. So here you can see the sample is up. Okay. Now you can turn off the lift and this is done. So thank you for your attention. I hope you were able to learn something from this video about NMR spectroscopy and how to record typical NMR. Thank you very much.
Yeah, welcome back. So this video is basically explaining how to prepare the sample and how to run the sample, so how to analyze the sample. And of course, the interpretation part are to be discussed. Of course, we know that the volume was slightly low, but what I found is in the YouTube channel, you can find that it is quite audible. So those who have had issues, you can go back to our Department of Chemistry YouTube channel, and there the workshop contents are being live streamed. So there, this content will be available, so you can go and listen again if you are interested. So now we will continue with the data analysis. So basically, in the first part of this session, Dr. Nagarjuna has told about the importance of using deuterated solvents, or the need of deuterated solvents, and how do we distinguish the peaks which is arising out of this deuterated solvents. Okay, so now we will look at some of the typical spectrum, and how do we assign the peaks to a particular proton or hydrogen atom, that we will see in the next session. So if any queries are there, you can just type in. Oh, there are some. Yeah, only the. Yeah. Yeah, I think Dr. Nagarjuna.
Yeah, in generally, actually, it's 0.5 ml is sufficient. It is, but sometimes we need solubility. Some compounds may partially soluble. In such case, it can take some little excess. And another thing for selecting solvent, we need to try also with some normal, like for example, if it is not soluble in CDCl3, then you have, suppose in DMSO-D6 means before using DMSO-D6 solvent, you can first check with DMSO, normal DMSO solvent. So then you can, if it is there solubility you observe nicely, then you can go for DMSO-D6 directly, because otherwise it is a waste of that solvent if it is did not solve.
Yeah, is one
Yeah.
more question is there. That is, which one is the best solvent for lipopolysaccharides? Any?
Right now, because we need to see the structure, like what kind of functional groups and all, because it depends upon the functional groups. So if you have a more functional groups, more polar functional groups, definitely you need to use polar solvents. So it entirely depends upon the functional groups.
Can we do solid state NMR? Yeah, we have a session coming up on solid state NMR. Ayan Ghosh, you may stay tuned. So today we are only discussing about the proton, and then later we have sessions on solid state NMR. So there we will discuss or we will address this question. Okay, then Dr. Nagarjuna, there is one more question.
Yes, sir.
When we add the sample of which we have to analyze, Kamini Patil, can you just elaborate the question? It's not clear. Kamini Patil, either I can unmute you or you can type in. Yeah, okay. You want me to unmute you? Yeah, maybe the control room, can you unmute?
Yeah, I think they're asking we can add TMS solvent. It's not required actually. The purchasing, while purchasing only, they will add some trace amount of TMS.
Yeah, there's TMS spike is always most of the samples solvents it is already spiked with the TMS, and nowadays the machine has the capability to identify the zero peak based on the deteriorated solvent we choose, because before starting the experiment itself we have to say whether we are using a CDCl3 or we are using a DMSO. And as Dr. Nagarjuna has shown, each solvent has a very specific peak. Okay, so ppm absorption is there. So that the instrument will identify it, and then it will calculate the further. I mean, the identification of the zero peak would be identified based on that one. So that is in nowadays the TMS spike is not required unless it is for some very specific applications. Okay. Yeah, and then one more question is. It's no. When sample is added in solvent. So that I think the initial part of the video was about that only. Generally, the sample is taken in a small vial, and then we add the solvent into the sample. And we can, if you have a vortex mixer, you can use it for simply dissolving that solvent into so that sample into the solvent. I hope this answers the question. Yeah. Any more questions? Yeah. Okay. Thank you. Thank you. Any questions? Is there anything coming on the YouTube?
No, sir.
No questions are there. Okay, so we can move to the second session, second part of this session. So yeah, you have the presentation right, sir. Dr. Nagarjuna, you can continue the
Yeah,
presentation.
it's visible, sir. It's visible.
No, it's not visible. Yeah, it's coming.
So already we have seen the detail in preparation of the sample. So that is about in the recorded sample in Bruker. What in our department 600 megahertz geo. So the first I will discuss.
You make it in the presentation mode.
1 minute. First, now I will discuss. These are the type of compounds like we can see carboxylic acid, aldehydes, aromatics, and this is the various type of molecules. And we can see the ppm levels different compounds come in different way because it depends upon the shielding and de-shielding effect. The more de-shielding protons come in downfold and downfield, and then which are not more de-shielded, it will come in upfield. So now we will see some of the examples how it will you can see in the spectrum. So then first I will show the compounds, and then I will show the spectrums. Then I will show the how to analyze and how to do in the GeoL software. So these are some typical molecules. 2-iodobenzoic acid and this paramethoxybenzoylate and hydroquinone, toluene, and this is one of the drug, and some of the ammonia salt and ester compounds and some cage compounds. So we can see details. Like for example, we know carboxylic acid. These are rough values we can get from the ChemDraw. We can estimate like where the possibility of the values will come. So then we can, after recording, then we can see. For example, if you see this, the proton NMR 2-iodobenzoic acid. Generally, if you see, we used to get OH peak, but unfortunately, in CDCl3 we could not observe. But other compounds we can see nicely doublet and triplet other protons. But the same compound, if you record in DMS46, you can see nicely that acidic proton, that carboxylic proton separately, because these are remaining all 4 here it is to come. This is entirely because when you are changing the solvent, CDCl3 is a very nonpolar solvent. Sometimes the compounds may solve partially, but whereas in DMS is a very polar, it will nicely bind with the compound, and especially may form with hydrogen but with the acidic proton. So that's what we can easily separate. We observe in DMS46 this kind of acid form, and then we can see this kind of this is like a aldehydes. We can see this doublet of doublets, and this aldehyde peak will come around 9.8, and then methoxy it has come around 3.83 because the methyl group is attached to the oxygen because the more alternative atom, so it will be D-shield. That's what it will come in a downfield. And if you see another interesting molecule is this, I'm taking typical normal available compounds in the laboratory. This is a proton NMR hydroquinone. We know hydroquinone in terms of molecular oxygen, it will convert to diquinone. Generally, we start 1,4-dihydroxy. When I record in this in a proton NMR CDCl3, we did not observe any OH protons. We see only this these double bonded protons. We can see 4 in a single region. And moreover, generally aromatic one generally shift to more than 7. But since it is a hydroquinone, that's what we got around 6.74 protons. And the same compound, if you record in DMS, we can see nicely the 1,4-dihydroxybenzene. You can see here these are 4 corresponds to the these 4 protons, and then this OH compounds used to come here 8.56 because it is attached to the benzene. So we can see that by changing the solvent we can observe the different even compounds. And another thing, toluene. One of the toluene is very interesting molecule. If you see the number of books in NMR, they used to explain about toluene. In general, if you see the toluene, we used to get total 4 signals. See this methyl, it will come, it's a singlet peak around around 2.3. I already told when in the first case 4-methoxybenzoyl, when that CH3 is attached to oxygen or more alternatively, it was downshielded and it directly benzene CCH3. That's what it came around 2.387. But generally we have to get these 7.06 two doublets and then a triplet. Total 4 signals we have to get. But since when you go for a solution soluble in compound, and if you record, we may not observe the same pattern. It may change. See, for example, it is not exact triplet, but there is a slight twist in the peaks. So this kind of changes matter. The same compound recorded in even DMS resins also. Even though here also we do not observe, but compared to that one, we can see this kind of triplet, and this is doublet and minute triplet. So this kind of changes we can observe. For example, in DMSO, we can see one, and here this one set of compounds and the peaks, and this one doublet is one set and triplet. So this may in DMSO we can little resolve the this triplets and doublet. And another molecule, this one, if you see, this is celecoxib. This is a like pyrazole derivative. We can see methyl group. It comes here 2.37, and then NH2 5.03. Even though if you do in a ChemDraw, it was showing 2.0, but NH2 it's entirely sometimes it may vary. But other cases, the benzene and cases, we can get approximate of values on the ChemDraw. And whereas pyrazole one, it was little bit upfield. And then this is one of the interesting molecule. If you see, this is ammonium salt. So we can see nicely there is a broad peak. Most of the ammonium salts we can observe as in a broad. If remaining all are CH protons, and then this ammonia we can see nicely the broad 4 protons. Even though it's showing very low intensity, but the number of protons nicely matching. And this is one kind of an ester compound. We can see ester, the CH2 and CH3. We already know the splitting pattern. This CH2 will get quadrate, and this corresponding CH3 will get triplet. This one we can see nicely. For example, if you see, it's a very matching very nicely. The ratio also very nicely we can see in the proton NMR. Finally, this is one a cage compound. This is a symmetrical molecule. We can see this bridged CH protons. Here and here, to get here, we can get here 2 protons at around 8.18, and this is and remaining 4 CH protons, which 8.321. So this is recorded in S26. We can see this is the around 2.01. I already told the respect to solvent, trace of solvents of whatever solvent we are using, we'll get same. For example, if you see in DMSO, this 2.49 is the corresponding trace amount of CH3 as double bond was CH3, and this little amount 3.4 is the water. The other spectra also you can see 3.3 is the moisture peak, and 2.4 is the trace amount of CH3 as double bond O CH3. So these are the some typical examples I taken, and like simple further like acid allylic compounds and acetylens also will show peaks. Now we can see how to analysis in the by using software. Dr. Manju Varghese, it is visible.
Yes, yes, it is visible. You can proceed.
So this is the typical spectrum of 2-iodobenzoic acid. Then we can, by using this cursor, we can pull it down, and then with this one we can elaborate it. So then after that we can, by using this one, we can use. This is a GeoL software, and now we have seen Bruker. So those who are in NIT Calicut, it may be more useful. I think because Dr. Anujwar is purchasing GeoL software. So we can see here this is a triplet, and we can see a triplet of triplet nicely because this is we are having a 600 megahertz. That's what it is nicely separating the even triplet of triplet. And this is see sometimes generally 7.266 CDCl3. It may little bit vary, but we can, by using here we can select and make it to 1 also 7.26. We click, it will come on the exact 7.26, and then we can so after that you can integrate. So by doing analysis, we need to select some of the one, at least one peak. Generally, if you are having methyl, epoxy, or OH functionality, this kind of is there, then we know that methyl means 3. Then based on, if you give 3 and remaining all will be definitely it will come exactly what you are expected. But if it's methyl groups not there means, then in such case we need to first we need to know number of protons in your compound, and based on that you need to give integration. Well, first if you give, it will give very huge number integration. Then you can do normalization by taking 1, and then now if you set one particular proton, you know that it will come only 1. Then automatically it will come remaining all. You can nicely. This is a nice triplet, and this is CDCl3. So we know we will no need to integrate the peak, and this one around 1.11, 1.8. It doesn't matter. You can keep. Sometimes we may not put exactly all are like a 1. It will vary some 1.16. No problem. Then after that, you could put down. Then you can remove the peaks line, and then by Control F you can put your experiment name. Then generally we used to we put in publications based on the highest downfield peak. Suppose you are having 8 point, and then we can keep up to 11 to around 0. This is the standard pattern. We will show NMR generally in the middle of the peaks, which is the highest, and this is the typical. Then we can save in a PDF. So this is the 2-bed angel. Already I showed you there is no OH peak. Even though I raised the intensity, I could not observe any OH peak in the OH peak of benzoic acid when I record in the CDCl. So I told you it's maybe because of CDCl, it may not interact much with the compound. So that's why we could not observe the benzoic acid. But now if you see, it is visible, Dr. Anujwar.
Yes, it is visible.
Yeah, okay. So now if you see the same 2-iodobenzoic acid, when we recorded in DMS, we can say nicely the peak acid peak around 13.3, whereas in CDCl3 we could not see. So this kind of sometimes we need to be checked with the different solvents to know the exact peaks. So then, and similarly other are not same. And then now I will go for sample another 2. This is 4-methoxybenzaldehyde. So already I told. So we know that benzaldehyde only one CH peak. Then you can select this one and give if you want, and remaining all we can nicely we can put it. Then we can see, because it's a 4-methoxy, we can see simply nicely doublet of doublet. So again, I told you the solvent, we need to select it. And then picking, you can select solvent. So I told you 7.24. Then again, you can adjust with the taking this and then 7.26. The remaining all you may change. So this is the spectrum of 4-methoxybenzaldehyde. And then we can see this. I told you hydroquinone when we recorded in. You see, this is you can observe here the by looking the spectrum. Even though I added more compound of 2-hydroxyquinone, but with the solvent peak is showing higher. That means definitely it's the partially soluble. It's not completely soluble. That's what we got more amount of CDCl, and the intensity of CDCl is more. Definitely, it's a partially soluble. That's what here I told you. That's got instead of 1,4-dihydroxybenzene, we got 1,4-diquinone compound. That's what the remaining 4 CH we are 6.6. In the same compound when recorded in DMSO, you can see nicely here this one corresponding to OH photons, and this is corresponding to the 4 CH photons. And I already told you can see this DMSO whenever you are using, whenever you are seeing in your spectra around 2 point. Second, we can adjust this one around 2.46, around 2.5 adjust. And then if you see 3.3, 2.5. Whenever you see any spectra, the compound, if you 3.3 and 2.4, you can definitely you can say that it's the compound was recorded in DMS or D6. Then another I told you toluene. So this is toluene. You see this one. This methyl protons. It's very important. It is very easy to whenever it is methyl is there, it is very easy to analysis the compounds, because we know methyl means 3 protons. Directly, if you give these three protons and remaining all, easily we can select it. We can assign very nicely. And remaining all, then I told you, toluene generally in the books we will see that two doublets and 2 triplets. So, but when you record, it is not exactly, because when you are taking a solvent, it may little recorded when it may changes occur. Here we can say that it's a triplet and it's a triplet and it's a doublet, little, but not exactly. Yeah, if you see this compound, even though this compound, whenever directly when you see spectra, sometimes we feel difficult to assign which one is a complex molecule. But already we know there is a methyl proton in this compound. So then it is easy to, again you select, and if you give 3, and then remaining all will be definitely it will match. So such way you can reduce your time also if having a in our case of methyl functionalities. And Other molecule, this one, if you see this glycoester, this one I told you already, aliphatic, most of the aliphatic will come in upfield. This NH, this is that this NH little broad and because it has a symmetrical molecule and corresponding in carbonyl groups, that's why it came very high region downfield. And then if you give this 4 protons, so sometimes ethyl acetate also used to come impurity. Sometimes people will think that my compound has formed, but you need to be careful whether it is your expect it is in the compound peaks or it is in an ethyl acetate peaks. So in such case, you have another because I know 4 NH has to come accordingly. Then I will select here. You can see nice quadrate. This nice quadrate we can this very nice quadrate. Can elaborate and you can keep this one in spectrum, middle of your spectrum also when you have publication. And this is the triplet. You can see this is nice triplet because CH2 CH3, we know CH3 will take triplet and CH2 will come quadrate. So we can assign like here, and then we can save it. And then another typical that salt compound I told you. This is also interesting molecule. So even though you see intensity is very low, but definitely it has to be 4 because I know in my compound ammonia salt is there. So then I don't bother about whether these all are matching or not. First, if I set it is 4 and it has to match other compounds with the respective protons. If it did not match, definitely it is something maybe whenever you add ammonia, it was not react with your compound. So in such case also, because I know these are only 3 protons, then definitely it will come. I have to get only 3 protons. Here 1, here 2, here 1. So I have 4 CH carbon protons, and I know salt because I taken simple NH2 compound. I converted to salt. How do I know confirmation in NMR means that earlier case if I give 1, it shows corresponding 1, 2 and 1. But now I given 4, it means it has showed exactly matching the other CH product. So like that you can give confirmation. And I told you already most of the NH2 or NH will come as a broad. And after that you can save. And lastly this cage compound. So this is I told you already simple because of symmetrical molecule. You used to see the intensity is a and 2:1 intensity because 4 protons this correspond and this is corresponding to 2 protons, and we can see I told you already acetone. We can see this what acetone when record it will come around 2.0017. These are the like that we can assign. First, generally, while in analysis, first we need to select the solvent properly. We need to set it. If you have any little change means, then with the spectrometer and with already in the instrument already they calibrated, then you can select this one. And then if you make it to this one, automatically there is exact value like 7.26 or 2.05. Like that, the stone solvent peak we can assign exactly. Then other one change. Sometimes maybe because of chimming and not proper, there is some kind of variation of your the solvent peaks. So this is one. This is the major point I want to discuss.
Yeah, I think
Yeah,
if
so I want to thank you all for your kind attention. Yeah, if any questions is there,
yeah,
we can discuss.
so if participants have any questions, they can ask. Yeah, I think maybe I anticipate some questions from NITC, our students. No questions? Okay, there is one question. Dr. Nagarjuna?
Yeah, it is for this question there is no answer. Which one you are practicing daily? That will be. See, earlier we used to use broker, and when we come to dual software, it's very difficult. But now we are habituated. So it depends upon your practice. Even though this mesh now also we can use that also they have a lot of good options. But we need to practice which one you are practicing daily. That will be best.
Yeah, one more question is there.
Yeah, generally volatile means because there is a option of in NMR you can study low temperature study also. So if you know that your compound is volatile, it need to be immediately recorded in low temperature. That may be solution. Because NGOL and I think in broker also there is a low temperature. Up to minus 80 can go for recording the NMR.
Okay, if there are no more questions, then I thank Dr. Nagarjuna for nicely explaining this. Of course, starting from the selection of solvent, the analysis of the data which is obtained and how to assign the peaks and all. So we will continue this session in the oncoming sessions like on Friday and all. So there, here we have discussed mostly about the proton NMR and to how to analyze the proton data. And there we would be mostly concentrating on the C13 data and as well as N15 data. So how to collect the data and as well as how to analyze this C13 and N15 data would be discussed in the next NMR data analysis session. Okay, so with this, I request the control room to take over the session.
It was an interesting session. Thank you very much, sir, for sharing the skills. Dear participants, today's next session is scheduled at 6 p.m. in the evening. Kindly be online at 6 p.m. for the last session of the day. Thank you.
Thank you, madam.
Thank you.
Hello, Professor David Bryce.
Hello.
Hello. Myself, Parameshwaran.
Hi.
Yeah, hi. I'm going to chair the session. Are you ready?
Yeah.
Okay. So, control room, we can start the program.
Welcome back to the last session of the day. Our honorable speaker is Professor David Bryce from University of Ottawa. He'll deliver a talk on basics of solid-state NMR interactions and applications to the study of non-covalent interactions. It's my pleasure to welcome Dr. Parameshwaran P., Associate Professor, Department of Chemistry, NITC, to chair the session. Welcome, sir.
Thank you. So, very good morning to David Bryce, and of course, good evening to our colleagues and participants. Certainly, it's indeed a great pleasure for me to introduce a very eminent and a very distinguished speaker, Professor David L. Bryce. He did his PhD from Dalhousie University in 2002. Then he moved to National Institute of Health for his postdoctoral work. Currently, he is a professor and chair of Department of Chemistry and Biomolecular Science. He is currently holding many reputed positions, which include University of Ottawa Research Chair in Nuclear Magnetic Resonance, Editor-in-Chief of Solid-State Nuclear Magnetic Resonance, Section Editor of Canadian Journal of Chemistry, Editorial Board Member of Chemistry Open, Spring Nature Applied Science, Advisory Board Member of Research in Chemistry, and many more positions. And he has received several honors. Some of the selected honors are Fellow of Royal Society of Canada, Fellow of Royal Society of Chemistry UK, Keith Ladle Award in 2014, sponsored by Canadian Society for Chemistry. His research interest includes solid-state NMR studies of halogen bonds and other sigma-hole interactions, novel effects and application in solid-state NMR spectroscopy of quadrupolar nuclei, NMR crystallography and polymorphism, and many more. And with this short introduction, I invite Professor David L. Bryce for his lecture. He is going to present on the topic Basics of Solid-State NMR Interaction and Application to the Studies of Non-Covalent Interaction. Over to you, sir.
Thank you very much for the introduction. I'm assuming you can hear me and see my screen?
Yes, certainly, yes. No issue.
Okay, great. Well, thank you again for the introduction and for the opportunity to speak at this workshop. It's a pleasure. Good morning, good evening. Yeah, I'm going to talk today about, well, more related, I guess, to the analysis aspect of this workshop, rather than describing in detail the theoretical basis of all the NMR interactions. I'll talk a bit about the analysis aspect. And so the first half of the talk will be largely educational, and then I'll present some research results on non-covalent interactions in the second half of the talk. So yes, I'm based in Ottawa. It's about, I think, about 13,000 kilometers from where you are. So nice to be able to make good use of these online tools these days during the pandemic. We just had an election, federal election last night in Canada, so I was up late, but that's all done now, and it's a pleasure to be with you this morning. Ottawa is a nice place. It gets 4 seasons. All 4 seasons. So right now it's cool in the mornings and warm in the afternoons as we're moving into fall. And I'm based at the University of Ottawa, right here in downtown Ottawa. So as I say, the first half of the talk today, I want to talk a bit about NMR interactions, and in particular, I guess, the analysis, line shape analysis of these interactions in solids. So looking at both isolated spins, so spin 1/2 nuclei, and quadrupolar nuclei, which are spins greater than 1/2 and make up about 75% of the periodic table. And then isolated spin pairs, where we have either 2 spins 1/2 2 quadrupolar spins, or one of each. And then in the second half of the talk, as I mentioned, I'll give you a couple of research results looking at some non-covalent tetraol bonds. Okay, so I'm not 100% sure of the background of the audience, but I'll present briefly here the kind of the 4 main interactions, external interactions that we consider in NMR of solids of diamagnetic materials. So most of you, I'm sure, are familiar with the chemical shift and the J coupling, for example, which are the main parameters we look at in solution NMR. So in the solid state, they're also important. So the magnetic shielding interaction is the fundamental interaction that shields the nucleus and which gives rise to the chemical shift that we observe in solution and solids. The J coupling, otherwise known as the indirect nuclear spin-spin coupling, is probably familiar from solution NMR. It's a coupling mediated by intervening electrons. And the solids, we have a couple more interactions that are often important. The direct dipolar coupling between nuclear spins. This basically averages to 0 in solution, but can play an important role in solids as it's dependent on the inverse cube of the distance between the spins, so we can get a lot of valuable geometric information. And finally, the nuclear electric quadrupolar coupling. So as I mentioned, 75% roughly of the elements in the periodic table have nuclear spin quantum numbers greater than 1/2. And so we have this quadrupolar coupling between the quadrupole moment of the nucleus itself and the electric field gradient at the nucleus, which is generated by the surrounding molecular and crystallographic structure, essentially. So I'll touch briefly on a couple of these interactions before getting into how we actually analyze NMR line shapes attributable to these interactions. So the first is nuclear magnetic shielding, which I said results in the chemical shift. And the shielding, essentially, the electronic structure of the molecule we're looking at modifies the apparent magnetic field experienced at the nucleus. So here we have our main applied magnetic field. We have some sort of electronic structure in which our nuclear spin is embedded. This modifies the apparent field felt by the nucleus. We don't measure this directly by NMR, as I've said a couple of times now. Instead, we measure a relative shift. We use a reference compound. And theoretically, this is often described using the Ramsey formalism shown here. So we have a diamagnetic and a paramagnetic contribution. I won't walk through these, but the important point here is that these are intimately related to the electronic structure of the molecule you're looking at, or the chemical bond you're looking at. And so if we do a proper analysis of the complete magnetic shielding or chemical shift tensor elements, often in concert with quantum chemistry, we can get a lot of valuable information on electronic structure. Now, this magnetic shielding tensor or chemical shift tensor has up to 9 independent elements, actually, and you're probably familiar mainly with the isotropic components. So the isotropic chemical shift is what we would see in solution NMR. And if we do fast magic angle spinning in the solid state, we also recover the isotropic chemical shift. So if we have a powdered sample, which is what we normally look at in solid state NMR, you could consider, for example, this labeled carbon-13 atom in phenylacetic acid here, which is marked with a star. If you imagine turning this molecule at 3 orthogonal directions relative to the external magnetic field, you actually get kind of the 2 extremities here, which we call delta 1,1 and delta 3,3, and then some intermediate value delta 2,2. That is, the chemical shift actually changes with the orientation of the molecule in the magnetic field, and in a static powdered sample orientation, so you map out this powder pattern in blue. So each kind of crystallite in your sample gives rise to a different peak, which gives us the complete powder pattern. And if we do magic angle spinning, so rapidly spinning our sample about an angle of 54.74 degrees with respect to the magnetic field, you can turn this powder pattern into something approaching a high resolution spectrum, where the faster you spin, you recover the series of what we call spinning sidebands, depending on how fast you're able to spin. And you can see if you spin more slowly, you can have both high resolution and still recover the 3 tensor elements. The other interaction I wanted to touch on briefly here is the nuclear electric quadrupole interaction, because as I said, a lot of elements in the periodic table are quadrupolar spins. This is an interaction between the nuclear electric quadrupole moment of the nucleus, So the charge distribution within the nucleus is non-spherical. It interacts with the electric field gradient. It's also orientation dependent, meaning that it's characterized by a second rank tensor. So if you have a crystallite and a solid, the response will be dependent on how your molecule or your crystallite is oriented in the magnetic field. It's typically characterized by the quadrupolar coupling constant and an asymmetry parameter. And one of the reasons that we're constantly striving for higher and higher applied magnetic fields in NMR is because this quadrupolar broadening, the spectral broadening introduced by this interaction, is reduced in Hertz as we increase the applied magnetic field. And I'll show some examples of that later. So if you consider a spin 3/2 nucleus here, we start with the basic Zeeman interaction, which splits the nuclear spin energy levels. And then if you consider the quadrupole interaction, basically these energy levels are further modified. And so you get not just a sharp peak at the isotropic chemical shift, but you get what we call the central transition here, 1/2 to -1/2, and then these broader satellite transition powder patterns. So we can get quite broad patterns due to this much sharper than the satellite transitions. So if you look here at some simulated spectra for that central transition, you can see the shaded spectra represent what we would get for, let's say, a spin 3/2 nucleus, an isolated spin 3/2 nucleus for a powdered sample with no magic angle spinning. So you have this broad pattern which has a characteristic line shape depending on the asymmetry of the tensor. So you can't get rid of this broadening. And if you do magic angle spinning, that will narrow that pattern by a factor of about 3 to 4. That's what's shown in these transparent or clear traces in the middle. And the breadth of this central transition, you can see here, depends on a variety of factors. So it depends on the Larmor frequency. So that will obviously be influenced by the applied magnetic field we're using. So the higher the magnetic field, the higher the Larmor frequency, the narrower the line width. These lines are also narrower for higher spin quantum numbers. So for spins 5/2, 7/2, 9/2, they're not as broad as for spins 3/2. And this is also mainly determined by the strength of the quadrupolar coupling. Okay. So I want to get in now to actually looking at some of the line shapes that we would analyze that are attributable to these interactions. So let's start with an isolated spin. And what I mean by an isolated spin is you have something like a carbon-13, which for practical purposes is not coupled to any other spins. So perhaps you're doing proton decoupling, but there's no other abundant spins nearby. So there's no dipolar or J couplings to this spin. We're going to look at what happens under stationary conditions for a powdered sample, so with no spinning. And then what if we do magic angle spinning, and talk about the symmetry elements that are relevant. So if we consider an isolated spin one dimension, it's characterized, if we consider chemical shift, by 1, 2, 3, what we call principal components, delta 1, 1, delta 2, 2, delta 3, 3. The average of these three gives us the isotropic chemical shift. And there's a couple of other conventions in the literature here that I've noted here. So I feel that simply listing the three components is a transparent and easy way to understand what we're talking about, but there are other ways to describe this too. So for example, the span of the tensor is simply how broad the line shape is for this chemical shift powder pattern. As I say, there are other definitions, and one has to be careful about if you're looking at a paper and you're not sure what convention they're using, it's very important to look that up and be sure so that you're describing these tensors in the right way. Now, one of the interesting things about why we would characterize this tensor rather than just the isotropic chemical shift is because this can give us information on the symmetry at the nucleus. This has applications in NMR crystallography. So for example, in restricting the point group of the molecule that we're studying, or the space group actually that we're looking at crystallographically. So for example, if you have octahedral or tetrahedral symmetry at the nucleus, that's high symmetry. So in fact, the tensor is completely isotropic. Whereas if you have C1 symmetry at your nucleus, all 6 parts of this components of the symmetric part, the tensor, are different. Okay, and then somewhere in between, you can see the number of independent components that you measure from NMR can restrict the possible point groups, and you can go further and restrict the possible space groups. Now, what if we do magic angle spinning for this isolated spin 1/2 nucleus? As I showed you, as we start spinning, the broad powder pattern then separates as we spin faster and faster into the isotropic peak and a series of spinning side bands. If you want to analyze chemical shift tensors this way, fit the data. So there's a few good papers in the literature describing kind of the optimal conditions. So for example, this paper found that you want at least 5 spinning side bands with good signal to noise to be able to fit these data reliably. Often the slower you spin, the better, because you have more spinning side bands to fit. Just something to pay attention to. You can get these from a static line shape, or you can get them by analyzing the spinning sideband pattern, but you need to be careful. Okay, so that's isolated spin 1/2. You can basically have a static powder pattern. You can pick off the 3 principal components, or you can do magic angle spinning and analyze the spinning sideband pattern. There's only one interaction that we're considering there, the chemical shift. If we move on to an isolated quadrupolar spin, we consider now, in addition to the chemical shift, we have to consider the electric field gradient, which gives rise to the quadrupole interaction. And so we basically are going to have 2 interactions contributing to the spectra at the same time. So we're going to have to worry not about just about the magnitudes of these tensor elements, but also the relative orientations of these tensors. And those can be described by a series of angles. We're going to look at both static and magic angle spinning conditions, and I'll talk a bit about some other things that pop up due to the quadrupole interaction, namely a second order shift that we have to consider, and the influence of the satellite transitions, so beyond just the central spin 1/2 to -1/2 transition. So there's a lot of information on this slide. I just wanted to lay out the parameters that we have to consider here for an isolated quadrupolar nucleus. So the quadrupolar coupling constant, which I've mentioned, it's simply a product mainly of the quadrupole moment of the nucleus, which is a constant for a given isotope, and then this VZZ, which is the largest component of the electric field gradient tensor. So this is what's going to vary from sample to sample and give you some information about your molecule or about your crystal structure. And then you've got some fundamental constants here. This is the asymmetry parameter, and these are some other derived parameters. I mentioned already that we can narrow this powder pattern by a factor of 3 to 4 with magic angle spinning. And this is just a table here showing actually that if we consider satellite transitions, we can actually get much narrower lines than what we get for the central transitions. I'm not going to walk through all these numbers here. Normally we look at the central transition because it's narrower under static conditions, but if you're able to do fast magic angle spinning, sometimes you can get spinning sidebands for the satellite transitions, which provide higher resolution. Okay, so I've showed this picture on the right before. I just want to emphasize again that if we look at the central transition and the satellite transitions, the satellites are quite broad. If we zoom in on the central transition, we have to treat this as second order, and we see it's actually not just a sharp peak. It has this characteristic line shape, which I've described here. And we can fit that line shape using software, which I'll show you, to get out an asymmetry parameter. And these also relate to things like molecular symmetry and crystallographic symmetry in similar ways to what I showed you for chemical shift tensors. There's a second order quadrupolar shift we have to consider. Again, I won't walk through the whole table here. Perhaps the best thing to look at is on the right here. And the point here is, if you consider these are simulated spectra here under infinitely fast spinning. And what you see is that under these conditions, 0 represents the position of the true isotropic chemical shift of this quadrupolar nucleus. But what we see is that actually there's no peaks that show up at the true isotropic chemical shift. They're all shifted to higher or lower frequency. So here's the central transition, spin 1/2 to -1/2. It's shifted. And this is due to this so-called second order quadrupolar shift, which depends again on the quadrupolar coupling constant and the Larmor frequency. And so this is something to be careful of when analyzing spectra of quadrupolar nuclei. The peaks that you see are never going to be at the true chemical shift. You have to actually use some software, ideally, to fit the spectra to take this second order shift into account. Now this effect can be diminished by going to a higher and higher applied magnetic field. So another reason for we're getting bigger magnets. So at higher magnetic fields, not only do you get narrower lines due to the reduction of the quadrupolar interaction in Hertz, but these lines also move closer to true isotropic chemical shift. So on the left here are some boron NMR spectra of solid hexamethylborazine acquired on a 200, 400, and 800 megahertz spectrometer. So we have experimental and simulated spectra here. And you can see the actual true chemical shift is actually marked here by this line here. So by going to higher and higher applied magnetic field, you can see the lines get narrower and they move closer and closer to the true chemical shift as we reduce the impact of these quadrupole interactions. It's the same under static conditions here. This is lanthanum-139 spectra of powdered lanthanum trichloride acquired at 5 magnetic fields. And again, as you go to higher and higher magnetic field, the lines get narrower and approach the true isotropic chemical shift. So effects are reduced but not eliminated by going to higher magnetic field. Now when we get into very strongly quadrupolar nuclei, that is where this quadrupolar interaction or quadrupolar frequency becomes comparable actually to the Larmor frequency or comparable to the Zeeman interaction, then things get quite complicated and we can't actually use second order effects to simulate these interactions anymore. We have to use an exact diagonalization of the combined Zeeman quadrupolar Hamiltonian, and I won't walk through all this, but the spectra can get complicated in this intermediate regime, and you have to be careful to use the right software to analyze this. So we've published some software back in 2012 which can do this. And of course, as we move to more and more strongly quadrupolar isotopes, we get into the regime of actually just doing nuclear quadrupole resonance spectroscopy rather than NMR spectroscopy. So in that case, we get sharp lines that are determined only by the quadrupolar frequency, which are not centered about the Larmor frequency. So there's a variety of options when dealing with very strongly quadrupolar nuclei. So again, sticking with an isolated quadrupolar spin here, I've talked a bit about the quadrupolar coupling, but we have to remember that we also have chemical shift and isotropy at the same time. Okay, so a given quadrupolar nucleus will be influenced both by the quadrupolar coupling and the chemical shift at the same time in the same spectrum. And these are some simulated spectra here, just showing the kind of results you can get depending on the tensor orientations, depending on the site symmetry. So it's highly orientation dependent, and in fitting these types of spectra, you have actually up to 8 adjustable parameters because you've got 3 tensor magnitudes for the chemical shift tensor, you've got 2 magnitudes for the quadrupolar coupling, and you've got 3 angles to adjust. So again, this can be challenging. It's worth acquiring data at multiple applied magnetic fields because of the field dependence of these interactions. But if you can do this type of thing, you get a lot of direct information, again, on site symmetry relative to each other. And these tensor orientations, again, are dictated by essentially the molecular and crystallographic structure. Basically what we're looking here, I'm going to walk through this so-called ZYZ convention, which tells us how we define these 2 axis systems. And this is one of the conventions that's often used. And there are 3 angles that ultimately define these orientations, alpha, beta, and gamma. So in this case, if you have a black axis system and a red axis system, the alpha rotation would be you hold the black z-axis, rotate the red counterclockwise about the z-axis by an angle alpha. That's the definition of alpha. You then hold the y2 axis fixed, rotate counterclockwise about that one. That would give us this beta angle. Finally, the gamma angle, you grab hold of the Z again and rotate a 3rd time. So these 2 axis systems here in black and in red are now, the relative orientations are defined by these 3 angles, alpha, beta, and gamma. This is something else that you need to be very careful about in analyzing spectra because there are different conventions in the literature. And there's a few pieces of software out there to analyze these types of interactions. We have one called EFG Shield. There's a couple of others, MagResView and TensorView. And again, why is this interesting, or why do we need to know about these angles? So this is something that comes out of spectral simulations. And what I'm showing you here is a series of simulations for an isolated sodium-23 spin 3/2 nucleus, influenced by both quadrupolar coupling and chemical shift anisotropy, and the relative orientations of the two tensors. And you can see you can get very different looking spectra depending on the site symmetry at the sodium. So again, tensor orientations are dictated by crystallographic and magnetic symmetry or lack thereof. Known crystal symmetry can reduce the number of unknowns in spectral fitting, or conversely, we can use the fitted tensor orientations to possibly restrict, for example, the allowed space groups or site symmetry. I won't dwell on this. Sometimes there's some confusion in the literature about tensor orientations. So I've described this Z, Y, Z order, and there's some pitfalls in the various conventions, but if you're interested in this, there's a bunch of useful references here and also some references on the previous slides. Okay, in this third and final part of the first half of the talk on kind of the educational aspect, I want to talk now about spin pairs. So what I've been talking about now so far is an isolated spin where you only have one spin basically interacting with the surrounding electrons, giving rise to chemical shifts or quadrupolar coupling. But what if we now have couplings between spins, so J couplings, for example, or dipolar couplings? So here we really have to consider the issue of chemical and magnetic equivalence in solids. So you may be familiar with this from solution NMR. If you have chemical equivalence, the kind of splittings that you might get in a proton NMR spectrum in solution. If you have magnetic equivalence, we know, for example, the 3 protons on a spinning methyl group in a molecule in solution just give rise to one sharp peak in the NMR spectra. You don't see the effect of the J coupling between these magnetically equivalent protons. Yes? Did somebody say something? No? Okay. Yeah, in solids we can consider, for example, AX, AA prime, and A2 spin systems. These have similar meanings as they do in solution NMR. So an AX spin system would be where you have, for example, either 2 different isotopes or spins that are not related by a symmetry element, essentially. You can then have chemical equivalence, AA prime, where they might be related, for example, by a rotation axis. But these are not magnetically equivalent. Magnetic equivalence implies we have identical tensor magnitudes and orientations. And this is represented by the A2 symbolism. And this typically is most easily visualized if you have, for example, an inversion center connecting your 2 spins. So completely magnetically equivalent. No matter how you change the magnetic field, these are always entirely magnetically equivalent. And this is important because depending on whether you have, for example, AX or A2 spin system, you get different NMR spectra again. So for example, here there's a particular splitting in these powder patterns, which can give you a dipolar coupling between the spins, the distance between the spins. But this distance is amplified by a factor of 3/2 if you have magnetic equivalence. So if you want to analyze these types of powder patterns, you need to have some understanding of what you're working with, or the spectra to be misinterpreted, and you get the wrong internuclear distance. Here's an experimental example showing how we get this distance. This is your carbon-13 spectra of stationary silver cyanide. And so what we're seeing here is in the carbon spectrum is coupling between the carbon-13 and the nitrogen-15. And the splitting on the end actually gives us the dipolar coupling between the spins. We can analyze this to get a distance between carbon and nitrogen. If we prepare the same sample again, but with N14 instead of N15, N14 is a spin 1 isotope, and so actually this splits into 3 rather than into 2. That's another piece of information that gives us the same distance. If we have 2 pairs of spin 1/2s that are actually form an AB spin system, that is, they're tightly coupled, so different chemical shifts, but still tightly coupled via dipolar and J couplings, things get a little bit more complicated. This is all programmed into software, though. There's an example here again for phenylacetic acid, where we have carbonyl carbon, which is coupled to a CH2 carbon, and there's data here acquired at 3 magnetic fields, and these spectra can be analyzed both using the chemical shift tensors and the dipolar coupling to get out internuclear distances. If you have tightly coupled spins, so AB or AA prime spin system in the solid, you can start to get some strange spectra, which actually depend on how fast you're spinning. So what I'm showing you here are N15 magic angle spinning spectra for doubly labeled cis-azobenzene dioxide, and you have some strange effects which show up here due to the interplay between chemical shift, dipolar, and J tensors. They're spinning speed dependent. And again, these effects are reduced but not eliminated by going to higher and higher magnetic fields. So there's a lot of things you need to be aware of or careful about, I guess, if you're trying to analyze solid state NMR spectra of spin pairs. And then the final part in this educational section is then looking at couplings involving quadrupolar nuclei. So if you have a spin 1/2 coupled to a quadrupolar nucleus, again, you get these second order effects, which are going to complicate the spectra. And they're going to depend not only on the quadrupolar coupling constant of the quadrupolar nucleus, but also on the dipolar coupling between them. So the distance between them, for example, and also on potential J coupling between them. So I'll show you an example here so you can see what I'm talking about. This is looking at triphenylphosphine oxide, which has been prepared with about 35% O17 enrichment. So oxygen 17 is a spin 5/2 nucleus. And so for the 65% of phosphorus in this, which is not bonded to an O17, we just get a sharp peak here, about 27 ppm. But for the 35% of molecules that have a phosphorus 31 coupled to O17, we get this pattern in the baseline here due to the J and dipolar coupling between phosphorus and oxygen. But this pattern is not a simple multiplet that you would see in solution NMR. We actually have splittings, separations which are farther apart at one end of the spectrum and narrow at the other end of the spectrum. This is called a stretching and squeezing, which is due actually to the dipolar coupling between the phosphorus and the oxygen. And this cannot be completely removed using magic angle spinning. Again, it can be reduced by going to higher and higher applied magnetic fields. So again, something to be very careful of. And there's a couple of other examples here. So for example, a cadmium nucleus, cadmium 113 is a spin 1/2 nucleus. In this case, it's coupled to 1, 2, 3, 4 nitrogen 14s, which are quadrupolar. But look at the complicated spectrum that results from this. Okay, it's not simple 1/2 nuclei. It's not what you would expect for coupling in solution. These quadrupolar effects really need to be analyzed carefully because there's this residual dipolar coupling between these elements. And of course, if we have all interactions contributing, so dipolar coupling, J coupling, quadrupolar coupling, and chemical shift, then you need to really analyze things carefully. You're going to want to acquire spectra at multiple magnetic fields. You're going to want to use kind of all the tricks you can to properly simulate these spectra and get out relevant information. If we have 2 quadrupolar spins coupled to each other, things get even more complicated. There are advanced techniques like multiple quantum magic angle spinning and double rotation NMR, which we can use to analyze these spectra. We actually showed student Fred Perra developed these methods that actually show if you have magnetic equivalence between a pair of quadrupolar spins, in this case 2 boron 11s, this magnetic equivalence shows up as an amplified J splitting in the NMR spectrum. And so it's a valuable tool that, for example, in this series of 7 boron diboron compounds, we can unequivocally show from NMR that in these 3 compounds there is no magnetic equivalence between the boron spins, that is, they're not related by an inversion center. Whereas these 3 on the right, the spectral splitting is amplified by this factor of 3, and this is a clear indication of magnetic equivalence. And of course, we can do this without knowledge of the crystal structure. This is quite a nice tool. I'll skip over this. We just did some similar work on gallium-gallium couplings, and we just see the same type of amplified splitting in the presence of magnetic equivalence. So a lot of what I've been talking about here requires appropriate software to simulate the spectra. There's a series of packages here that you can look at if you're interested. I think this is being recorded. You'll be able to look back and have a look at these software packages. Okay, now in the last part of the talk, I wanted to move into some experimental work from our group. So looking at a few recent papers where we've used solid-state NMR to study non-covalent interactions. So I'll give you a brief introduction to the idea of electrophilic non-covalent interactions in solids, the idea of sigma-hole interactions. And then I'll present some work on carbon-13 NMR of weakly tetrabonded methyl groups. That's been summarized in these first two papers, and then some more recent work on looking at tetra bonds to tin. If you're working in the field of crystal engineering, you may be familiar with the idea of naming interactions by the electrophilic site. There's a lot of interactions that are, I guess, being rediscovered or discovered in the literature these days. So if you think of naming interactions by the electrophilic site, the hydrogen bond is the classic example. You think of a hydrogen bond. In a hydrogen bond, the hydrogen is the electrophilic site. We call it a hydrogen bond. So all these other classes of interactions are named because the element in question acts as the electrophilic site, the electron acceptor. So if you consider, for example, the halogen bond, you can have, for example, a large polarizable iodine, which can develop a sigma hole opposite a covalent bond to iodine, and the iodine can actually act as an electron acceptor through that sigma hole. We've done a lot of work in this area. We're also working on chalcogen bonds and pnictogen bonds, so doing some, for example, antimony nuclear quadrupole resonance. We've done selenium and tellurium NMR. And today I want to focus on some tetrabonded systems, so carbon and tin. Now, so here's an example of what I mean by a carbon tetral bond. You see a methyl group here. In this case, the methyl is bonded to a nitrogen. If you could imagine an electron withdrawing group on this carbon, you can create an area of depleted electron density and elevated electrostatic potential opposite the covalent bond here. So that creates an opportunity for something like an oxygen to actually donate electron response. We also expect it to be weak because carbon has the smallest chemical shift range of the tetral elements. Nevertheless, we wanted to look into this given the ubiquity of methyl groups in a wide variety of systems. So can we detect tetral bonds by NMR spectroscopy? So my student Scott Southern looked at this system, some sarcosinium salts a few years ago, tried to look at the NMR response for a particular methyl group here. There's been a lot of computational work in this area, and so we wanted to look experimentally to see what we could see. And in this paper, using some computational work and some experimental work, where I would actually show a clear shift of a few ppm attributable to this tetral bond, so quite a substantial shift. We wanted to follow this up. We did this recently this year to see how general this phenomenon is and really how robust the effect might be, because as you can imagine, there's a lot of things affecting subtle shifts of carbon chemical shifts in methyl groups in crystal structures. So Scott looked at a series of co-crystals of caffeine and theophylline shown here. So basically some known structures that he found in the Cambridge database, and he prepared these. So here's an example of powder X-ray. And basically looking at tetral bonds to, for example, some of these carbon atoms. If we look at the structures here, you can see what I mean. So in turquoise, we have the purported possible tetral bond. These are all at distances slightly less than the van der Waals radii of the oxygen and the carbon. Only by a few percent, because again, we expect these to be weak interactions. But just to show you the kind of compounds that were prepared. You can see the types of geometries that we're talking about here. So distances, again, just slightly less than the sum of the van der Waals radii, so around 3 angstroms. And again, these often tend to be highly linear, so the angle concerning the tetral bond close to 180. And Scott recorded simple carbon-13 cross-polarization magic angle spinning spectra, so to recover the isotropic shifts for these compounds. So you can see there's in some compounds, there's several methyl groups, depending on the structure of the compound. He did some two-dimensional work. I won't walk through the analysis here, but we did some two-dimensional proton carbon spectroscopy to aid in the assignment of these peaks. DFT calculations were also useful in this regard because, again, there's a few different methyl groups in the molecule studied, and we had to be sure of the spectral assignments. I want to take a step back now from the experimental data to talk briefly about some computational work which was included in this paper. And what we saw is that DFT computations consistently show that this tetral bond results in an increased carbon chemical shift of the methyl group. So for all the compounds we studied here, what I'm summarizing in this table are the results of DFT calculations, basically for the single isolated theophylline or caffeine molecule versus what happens when you add in one interacting molecule of the co-crystal and the tetral bond is formed. And in all cases, we see this a shift in the same direction. So a decrease in the magnetic shielding constant of a few ppm in all cases, which translates to an increased chemical shift. So this is a very clear effect computationally. Again, the question is it borne out experimentally, or are there other competing effects that might drown this out? And what we see is what I would say are moderate to weak correlations experimentally. Okay, so here's the tetral bond distance. Here's the isotropic carbon chemical shift. There's a large degree of scatter here, but nevertheless, the experimental trend reproduces what we see computationally. In panels B and C, we have some computed chemical shift tensor elements, which also correlate a bit more strongly with the tetral bond distance. We also included some data from the literature from Vigilante and Mehta here, so the points in blue. So this is not just our data, other data seem to fit this trend well, but again, it's a moderate correlation. So to summarize this little section on weakly tetral bonded methyl groups, I would say both experimentally and computationally, the tetral bond does influence the chemical shift. As the tetral bond gets shorter, the isotropic chemical shift increases. As we saw from computations, this increase more strongly correlates with the distance in the absence of other competing structural effects. So when we can isolate the tetral bond, the trend is clear. In real systems, this seems to be weaker, as I'm saying in point 2 here. The magnitude of the effect varies, likely due to other competing crystallographic effects. But the effect is up to 5 ppm, typically a few ppm. In terms of the proton chemical shifts of the methyl groups, I didn't show you any of those data. There is a weak effect computationally, but experimentally the correlation.
I think he got logged out.
Yeah, what happened actually? There was some problem with the WebEx, it seems.
Is it?
Yeah.
Yeah, because everything collapsed, right?
Yeah, everything collapsed. Yeah.
For a moment. But still there are 80 attendees.
Yes, 84, including the panelist.
Yeah, no.
Hello?
It went back to 20 or something, and then it came back to 80 actually.
Am I audible?
Yeah, sir has come back.
Oh.
Hello?
Hello?
I think there was some issue.
Yeah, okay, I seem to be back, okay?
Yes, perfectly okay. Now it's okay.
Okay, great. I have one final section here, so that I'm not sure exactly when I cut out, but that was the end of the little section on carbon tetral bonds. And the last section is looking at tin tetral bonds. Tin is obviously a heavier tetral element. We might expect a bigger NMR response upon the formation of tetral bonds. And my postdoc Vijay Kumar looked at some crystal engineering applications with triphenyl tin chloride. So if you look kind of down opposite the tin chlorine bond, there's actually an area of depleted electron density and elevated electrostatic potential, which can act as an electron acceptor. So Vijay prepared a series of co-crystals with these electron donors. You can see the kind of quality of powder X-ray data we get, and some beautiful single crystal X-ray structures here, showing indeed the tetral bonding between oxygen and tin in this case. So these are highly linear interactions, and they are at longer than the sum of the covalent radii, but significantly less than the sum of the van der Waals radii. So here are some other examples with some different types of donor groups, using nitrogen or oxygen. So very nice structures and potential applications in crystal engineering. Here we actually see that the tetral bond still forms if you have solvent molecules. So for example, if the donor is perhaps too bulky to get in there, the tetral bond still finds a way to form with solvent molecules. Some interesting structures here. And then what I wanted to get into really here is the tin NMR. So in these types of systems, the tin NMR actually is a little bit complicated because tin is directly bonded to chlorine. Chlorine has two isotopes, 35 and 37. So the tin magic angle spinning NMR data that we're going to get are going to be affected not only by the tin chemical shift tensor, they'll be affected by the J coupling to chlorine, they'll be affected by residual dipolar coupling to chlorine, and by the chlorine quadrupolar coupling, as well as tensor orientations. So you need to be quite careful in analyzing these spectra. So they were acquired at a couple of different magnetic fields, so on a 200 and 400 instrument. These are the kind of data that we get. So in black is the experimental data, in red is the simulated data, which depends on a lot of parameters here. We're able to pin down actually the chlorine nuclear quadrupolar coupling data by acquiring the chlorine 35 spectra directly on a 900 megahertz instrument. So you get these very broad powder patterns. You can see here about a megahertz across. These are acquired in stepped frequency mode, and they can be simulated to extract chlorine quadrupolar coupling data independent of the tin data. Now for these compounds, the chlorine quadrupolar coupling constants are about 21 to 26 megahertz, and they actually correlate weakly with the tetral bond length. They're also quite different from the quadrupolar coupling for the pure triphenyl tin chloride. So these chlorine coupling, this chlorine coupling data is actually a reasonably good indicator of the tetral bond length. I don't have a graph showing that right now, but what I do want to conclude with here is this correlation that we've established, i.e. the J coupling between tin and chlorine actually correlates with this tetral bond length. So as you bring in an electron donor, it turns out this tin chlorine bond actually gets a little longer. The J coupling gets a little weaker, and there's a nice correlation here with a good correlation coefficient. So if we remove the tetral bond in one of the systems where it's quite long, the J coupling is quite large. As we bring in the tetral bond, this bond lengthens, and we get a nice kind of novel probe of tetral bonds. And we've backed up all of this work with a lot of DFT computations. I don't have all the results in this presentation today. And we've analyzed all the chlorine NMR data as well. So this is ongoing work. But it's kind of a nice example based on the first half of the talk of a case where you really need to consider all these interactions simultaneously to properly simulate your NMR spectra and get out some structurally interesting correlation. Okay, so I don't want to go on too long here. I will conclude for the second half of the talk by saying that electrophilic non-covalent interactions offer a wealth of opportunities for study by solid-state NMR because you have a whole range of interesting isotopes to study, and all the NMR interactions come into play. I showed you that methyl carbon chemical shifts tend to increase when engaged in weak tetraol bonds. And we definitely see a strong effect here on the J couplings between tin and chlorine, giving us a novel probe of tetraol bonds to tin. A lot of work in this area, looking at halogen bonds, tetraol bond, pnictogen bonds, and also some previous work on hydrogen bonds is summarized in this review, coordination chemistry reviews from last year. I will conclude here. Thank all the people in the group. This is from I think our first in-person group meeting in about a year and a half, recently in Ottawa. We were finally able to get together. And Scott and Vijith did most of the work on the carbon and tin work that I talked about today. And I'll thank you again for the invitation and for your attention, and I'm happy to take any questions.
Thank you, Professor David Bryce, for his very informative and a nice talk on NMR interaction, especially for understanding about non-covalent interaction. Now the session is open for discussion. Questions and comments, please. Yeah, there is one question in our chat box. Are you able to see, or should I read?
Pardon?
There is one question in chat box.
Okay, chat. I see Q&A. I'll look for the chat.
Okay, I can read that.
Okay, I see it. Yeah.
Okay.
So can we distinguish between chemical exchange? That one? I see a few questions here, so I'm not sure which one I should do first.
Yeah, yes.
In the chat box, I see, can we distinguish between chemical exchange and spin diffusion in solid state NMR?
Exactly. Yes.
I would say yes. I'm not an expert in those particular areas, but there are definitely, yeah, I think there would be ways to do that. I'm not an expert though on spin diffusion effects, unfortunately. How does solid state NMR of a crystalline sample differ from its amorphous version? Oh, good question. So yeah, I mean, one of the big pros of solid state NMR, certainly in comparison with diffraction methods, is that we can easily look at amorphous materials. So the basic difference is that your lines get a little bit broader in solid state NMR. So if you have a very highly crystalline sample, the line width will be limited by largely instrumental effects. But if you have an amorphous sample, this will be the lines will be broadened depending on the differences in environment across your more amorphous sample. So you can still generally analyze the spectra of such samples. There are actually quantitative ways to do that. And it's a very useful technique for being able to see even mixtures of amorphous and crystalline, or whether you have amorphous or crystalline material. Okay. Let's see.
Any other questions?
Okay, orientation of molecule is important. How NMR of powder sample and single crystal sample will be different. So if you have a single crystal, if you have, for example, one crystallographically unique molecule, let's say for the sake of discussion, in your unit cell, you'll get one peak in your NMR spectrum. So one very sharp peak, just like you would see in solution NMR. If you were to rotate that crystal, the peak will move, and it will map out the orientation dependence of the NMR interaction. So the peak won't show up, for example, at the true chemical shift. It will show up depending on the orientation of the molecule as well. If you have a powdered sample, now you have a bunch of tiny crystallites oriented at all possible orientations. And so now you basically get, in effect, a superposition of peaks from every tiny crystallite in the sample, and you get this broad powder pattern. And by doing magic angle spinning, you can then collapse that to recover the isotropic shifts. There are advantages to doing single crystal NMR as well, but the analysis is quite complicated. I see in the Q&A how to determine X atom other than H and C atom. Oh, maybe that's from a different talk. I'm not sure.
Any other questions from audience? I have one question. So in the case of hydrogen bond, right?
Sorry, I heard you say in the case of hydrogen bond, and then it cut out for a second.
No, why you are asking? Because you are talking about a tetra bond.
Yeah.
So is it a what type? Red shift or blue shift or both?
Sorry, I think I missed part of your question. It cut out.
Yes, in the tetra bond.
Yeah.
Is it a red shift or blue shift?
Oh, well, I mean, are you talking about the NMR or are you talking about infrared spectroscopy?
No, yeah, but in your presentation, you said something shortening of the bond. So
I mean, typically you would see let's say you have your carbon atom, you bring in, let's say, an oxygen. Yeah, the bond on the far side typically lengthens.
Typically lengthens.
Yeah, so typically you have electron donation into an antibonding orbital that will lengthen the covalent bond.
Okay, fine. Okay, thank you. Any other questions? Anybody raise the hand? No, I think, right? Anuj, anybody raise the hand?
No.
No, right? So if there are no other questions, let us thank the speaker on behalf of Department of Chemistry as well as all the participants gathered here. I express sincere gratitude to Professor David Bryce for his very nice lecture. Thank you, sir. Thank you very much.
Thank you very much for the invitation.
Okay. So now I will hand over the mic to control room.
Thank you, sir, for the wonderful lecture. Thanks to all participants for your kind attention and participation. With this, we come to end of day 2 of the online workshop. Hope all of you are having joyful learning sessions. Tomorrow on day 3 of the online workshop, we have 2 sessions each for lectures and NMR instrumentation. Please be online at sharp 10 a.m. Thank you. Have a good day.